these fungi, they are there everywhere. In Telugu, there is a poem. Indugala andaledan sandeyam valadu. Chakri endendu vedikina andande galadu. So like that,
सर अनम्यूट कीजिए सर, आपका चारी सर आपका साउंड नहीं आ रहा है।
अब ठीक है? आ रहा
है सर, अब साउंड आ रहा है।
So should I go for the first slide then?
हाँ सर, शुरू से कीजिए सर।
Okay, anyhow, these are the ecological niches on which the fungi will be present. Living vascular and non-vascular plants, and plant exudates, root exudates, and all that. And then dead plant parts, as I told you earlier, they are saprophytes, most of them, and they live on dead debris. Decomposition and waste recycling is an important part to keep the environment very much clean. And on other microbes also they are present. For example, chytrids are present on many of the algae, and also within many of the fungi also, within the fungi also you'll find this. And you see analogs also, and vertebrates, echinoderms and non-vertebrates also, that is echinoderms and vertebrates. Many of the skin diseases are because of, they are called keratinophilic fungi. They cause a number of diseases. Microsporidium gypsum, especially among the school children, it causes the disease. And histoplasma and Candida auris has created a lot of nuisance among the hospitals, and this has become a world problem. And like that, we have number of fungi which will be colonizing the hair and as well as the skin, and very difficult to get rid of these fungi from that of the human pathogens are very important. And they are present in water, air, soil, coprophilous, that is animal excreta, keratin, aeroaquatic habitats, water droplets, rhizosphere region, specialized ecological niche, and studies on rhizosphere. Rhizosphere has been 100 years old, and they are there on the leaf surface and rhizoplane on the root surface. In the case of compost, what the farmers used to do earlier is they used to keep the hay and other things in their fields for longer time, and they used to remove it and then use it as a fertilizer. So in that, number of fungi are there which help in the decomposition. A number of bacteria are also there, and all of you know that in the case of good olden days, the shepherds used to keep their sheep on the rice fields and other fields, and there they for about a week, and the droplets from that of the sheep used to be a fertilizer. In that also they could find, scientists could find a lot of fungi and the bacteria and PGPR, plant growth promoting rhizobacteria, and some actinomycetes also, which help in the soil's texture and soil formation, and also in the case of soil fertility. So these are some of the important aspects. And extreme habitats, like where the high temperatures are there, and where the thermophilic fungi do occur. In the case of Himalayan region, where the cold temperature is prevailing, there the psychrophilic fungi will be present. In the case of sulfur springs also, you will find. In recent times, recently means that is 1 week back, I have seen a paper that the bacteria have been reported, and some fungi, 1 or 2 fungi are reported on Mars also. So it is an important discovery. On your Twitter and also on your mobile phones, and many of the habitats, even if you shake hand, then also the microbes and fungi will be there colonizing. They may not be parasitic at the same moment, or they may not be pathogenic, but nevertheless, of course, now because of pandemic, now we are doing only namaskar, no shake hand at all. But nevertheless, all these habitats are there where you can find the fungi. And if you wash your hand after shake hand, you will find number of fungal spores and as well as bacteria. Now, It got stuck 1 minute, sir. So these are some of the things which I wanted to tell you. And ecological niches and substrates are very specialized ones. And now coming to the biodiversity, it is a variation among the living organisms, and it is a variation among the species, among the ecosystems, and it is a variation among the genera, and variation in physiology, biochemistry, and also in case of certain specialized ecological niches are there, in that also you will find variation. You will also find that among the genes also you will find the variation. Now that genomic editing and other things have come, barcoding and other things have come, the data is flowing, and you can see the right the front region, the top, that is soma, the fungus, the mushroom, where in the Vedic literature it has been mentioned that during the that munis and saints, they used to drink this particular drink liquid from extracted from this particular soma. It was a kind of tonic for them, and it is a mushroom which is now all of you know that Agaricus campestris and so on and so forth, Boletus, edulis, these are some of the edible mushrooms. Then second one is coral fungi. These do occur in the case of rocky regions and Himalayan regions and all that. This is an anti-cancer drug yielding fungus, very important fungus. And I think more than 30 metabolites, secondary metabolites have been secreted by this particular fungus. Lower down is the Odomantiella. In the night, this photograph has been taken in Vikarabad forest, where you can see that the luminescent basidocarp is seen in the night. And so what Mahatma Gandhi said is that the world has got enough of biodiversity for everyone needs, and for biotechnology, and for agriculture and so on and so, but not enough for everybody's greed. See, decline in the case of biodiversity, decline in the case of fungal population, decline in the case of many other woody trees and important medicinal plants is because of the greed and selfishness. And I think this is a very important message that one should take, that now coming to the beauty of the fungi, we think that our faces are only beautiful and we are only intelligent. But fungi are more intelligent, and they don't have tongue, twisted tongue like us. They don't have brain like us, and they will not speak. Very silently they work. They are silent soldiers, so as to say, in the ecosystem management, and they are also very beautiful. And whenever you find a new fungus, India has been a very strong center and cradle for fungi. Whenever you discover a new fungus, and you feel that very happy, and so much of joy you will enjoy. So mycologists, they have traveled many of the areas, and they have unraveled through conventional and non-conventional techniques. What they have discovered so far in this globe is only a fraction of Atlantic Sea only, a drop of it only, not the whole water has been screened, not the whole forest is screened, not the real diversity has been projected in India or elsewhere also. I will come to that point later on. And the first thing is that in the year 1967, Oxford said that biodiversity estimate, this is only an estimate, he said that it is 1.6 million. But later on, Hawksworth and Lucking in 2017, they said that it is up to 3.8 million. That means the estimate has gone up. And then now, Wu et al., along with few others, 2019, he said that the estimate what that has been done earlier is not correct. It may be 13 million of fungi. That means the estimate of the fungi is the second richest flora is the fungal flora after insects. Insects are number one, and the fungi are the second one. plants come later. So you think that you should think that the fungal flora, which is existing as a second largest biodiversity in India and in other countries, we should utilize them for the benefit of the human welfare. That we are not doing. So we think that our brains are very sharp. Though we have got many beautiful and useful fungi and useful microbes, we are not able to utilize them. Our brain cells are not that sharp to go and find out which fungus is more useful for us than other things. So now coming to the number of fungal species, estimate is 13 million, and now you have got 1,40,000 species are reported as on today from the whole world. Where is 13 million, and what is that we have identified? 1,40,000 only out of 13 million. See, it is again tells upon the capacity of the human mind. We are not able to identify, we are not able to collect the fungi, we are not able to go to the forest locality, we are not able to locate them. All these things are happening. So it is a big challenge to the human mind. And in India, there are 29,000 fungal species have been reported. Maybe India might have got around 3 million or 2 million of fungi. And to identify 13 million of fungi in 10,000 years are also not enough. And even to identify 2 million fungi which are going to occur that is an estimate in India, I think it may take more than 5,000 years also, because we don't have taxonomies to identify fungi, bacteria, plants, and other things. Hardly there are 20 mycologists in India to identify the fungi. Many of them have expired, and many of them have become old, and we don't have many of the taxonomies. So taxonomy is the mother of sciences. Everybody knows that taxonomy is very important. It is a way of life also. But nevertheless, whenever something new comes to us, then we will be after that only. When something molecular work model comes, then we will be after that. If somebody gives a project, we will leave our basic work, and then we will go to that particular work. So we are hanging upon the money that we are getting, and not on the knowledge and its dissemination. That is an unfortunate situation in this particular country. And out of 29,000 genera which are reported, 29,000 species, it represents 2,080 genera. And whereas 1,40,000 species which are reported from the world, it is represented by 7,270 genera. And this is a very important slide I have prepared for about working on for 5 years, and I getting the data from different research papers. Now this is also important. Out of 29,000 fungi, C.V. Subramaniam has discovered 32% of Indian fungal wealth. And then the 68% of fungal wealth has been discovered by many other mycologists. So we have described from Usman University, my students and myself, around 20 new genera, and then 120 new species, and 500 new fungi to India. And most of them we have reported, and some of them still we are publishing. We are in the process of publishing them. And where we should look for this particular one, the fungi, you should go to the forest, you should go to agriculture fields, you should go to wild environment, you should go to different places, even hospitals also, very good source for collecting the fungi, litter, and so on and so forth, many of the habitats. And niche identification, that is the substrate identification is very important. You collect it, and then underneath the binocular microscope, you see whether the fungi are there or not. Afterwards, you put them in the culture on agar media, make dilution plate, or make the soil plating, or make the litter plating, and so on and so forth, and rationalize all that and identify them underneath the microscope, high power microscope, and then identify the species level, characterize them morphologically, and also characterize them in recent days by means of molecular tools, and then come out with a proper identification. This is very important aspect that for the people who want to work on the fields, it can be, and they want to do some PhD or publish research papers, they have to see. So this is planning and implementation is very important. The another important feature is that the major concern is the decline of fungal wealth in the whole globe because of, as I told you, the jealousies and as well as the selfishness. If some fungus is very important, then people will be after that. Though it is not cultured, they will go on collecting it, and then by the time we want to see that particular fungus, it will not be there. For example, Marcella esculenta, an edible fungus called Gucci, is we are getting 4,000 crores by selling it to other countries. You show me any biotechnology product which is getting 4,000 crores. This is, and if you go on collecting, the mycelium will be lost, and that is not culturable also. That fungus, only mycelium they could produce in recent times, but never they could culture it and grow under laboratory conditions like button mushroom or paddy straw mushroom and so on and so forth. So this is a major concern. Conservation is very important. Protection of bioreserves or biosphere reserves is very important. Protection is to be offered. If you put a figure of a tiger in a forest and say that your fate is in your own hands, then nobody will enter into the forest. And fungi will be safe, and as well as many important organisms can be collected with the permission of the forest officials, and it can be utilized for the human welfare. Ex situ and in situ conservation. Most of the saprophytic fungi can be cultured, and single spore cultures are very important for biotechnology, for genetics, for physiology, for biochemistry, and so on and so forth. And whereas obligate parasitic fungi cannot be cultured, and because rust fungi also could not be cultured, powder mildews could not be cultured, albugo could not be cultured, sarcospora could not be cultured. So all these come under obligate parasites, and all these obligate parasitic fungi are to be maintained on living hosts only. And I will come to that point little bit later. Now, legal protection or codes of practice is very important. Inventarization of fungi is very important. We don't have inventarization. That means how many fungi are red listed. In India, we don't have any list. How many species are endangered? How many important fungi are declining? We don't have any data. So, with reference to the world, there are 16 fungi, 1,516 fungi are there. And in European countries and Czech country, they have developed a legal code. That is, legal code is that everybody has to take a permission from the forester, and they should collect only those fungi which are useful for them, and they should not go for other fungi. They should not destroy others. And if they enter without any permission, they will be behind the jail. They will be in the jail. So legal protection has already, that code and order has been passed in these countries. We don't have any computerized data. We don't have any mapping of the fungi in India, nor for the bacteria. Even for epidemic diseases also, we don't have data. We don't have any data for lead blood of potato, as Ireland was having and America was having, and in UK and other countries were having. We don't have any data on that. We don't have any data on rust fungi. We don't have mapping is not done. So database for biogeographic fungal base is very important. And red data book on endangered fungi. For example, Marcella esculenta is becoming endangered because of the collection, and mycelium is lost in the soils. It has been getting decomposed by other fungi and other bacteria, and it is declining because apple orchards, if you cut underneath the apple orchard, this fungus grows. If the apple plantation is not there, the fungus will not be surviving. That is the connection with the host. So if you lose one host, you will be losing many other important microbes and fungi in the rhizosphere region, and some macroscopic fungi like your Gucci. Now, these are fungal germplasm maintenance, lyophilization, and it is very important for us. And this is one day of the conservation of the fungi is very important, and we should go for that particular one. And lyophilization, freeze drying, mineral oil storage, water storage, soil storage, and so on and so forth, silica gel storage. And you have to deposit these cultures in national and international culture collection center. Now, Shanghai and as well as the Melbourne code, the botanical nomenclature, for fungi there is a separate nomenclature for some fungi, bacteria, and as well as for lichens. The Melbourne code is there and Shanghai code is there. They say that you have to deposit these cultures in two places. And you should deposit, if it is a gene bank, you have to deposit in two places. And if it is herbarium, you have to deposit in two places. So this preservation and deposition is very important. And fungal herbarium in India, we have got only one center, that is HCIO, the Herbarium Cryptogamy India Orientalis, Division of Plant Pathology IRI. This is the only center where fungal herbarium is accepted. Not in the case of Chandigarh, nor in the case of NBA Mau, nor in the case of Pune Culture Center and all that. They don't accept any herbarium. They accept only cultures. And Mau, they accept only agriculturally important organisms. So these are the culture centers. Now here you can see the National Bureau of Agriculturally Important Microorganisms. And I was the person to establish this particular one there. And now it is almost 20 years. It is now this particular center is accepted under Budapest Treaty. Now it is internationally recognized. Anybody from other countries also can deposit this particular one. And also the National Fungal Culture Correction Center, Agarkar Research Institute, Pune. Again, I was responsible to establish particular center at Pune under DST as a chairman of that particular committee. Microbial type culture collection, MTC Chandigarh. This is the oldest culture collection center. Industrially important organisms are deposited here. This is also accepted under Budapest Treaty. Is internationally recognized center. Indian type culture collection, that is IR in New Delhi. This is oldest center. And then now the fungi is that around 7% of fungi are only cultured. 93% of fungi are not culturable. They are not cultured. They are not cultured. As I told you, obligate parasitic fungi. We did not culture many of the lichens. We did not culture Sarcospora also. Bhama and Surnarayana at CS Madras, they have tried, but they could not get the whole culture correlation. Like that, some people are working, but till today, there is no pure culture. Hermetomyces, an edible mushroom, which grows on termite hills, could not be cultured. Marcella esculenta could not be cultured. Cordyceps sinensis, the India Viagra and anti-cancer drug is extracted from this, and it could not be cultured. So, like this, 7% of fungi are only cultured. Whichever is easy, that has been cultured. So is to say that only. And they are many are there which are not culturable. Our efforts must be there to culture the fungi so that biotechnology can develop in this particular country, fungal biotechnology. So there is a need to use all the techniques, all kinds of techniques, including molecular techniques. Now, classification of fungi. Classification must be easy, must be understandable, easy to follow. It should not confuse neither students, nor the teachers, nor the research scholars. It should not lead to any kind of complications. And this is the simplest classification, Saptora classification. And of course, earlier, Alexopholus and other things were there. Webster classification was also there. This is the recently one. And now, fungi has been accepted as a major kingdom. These kingdoms are Eumycota, Promista, and Protozoa. Why it has been accepted? Neither plant scientists wanted to include in their kingdom, nor the animal scientists wanted to include in their own kingdom, because the chitin cell is there in the case of fungi. Gregory, in 1971, he has published a paper. He said that is the fungi are animals because of chitin cell wall composition. And if the chitin cell wall composition is also there in the insects, then he questioned why animal scientists are not accepting. Microbiologists, they don't want to accept it. Only yeast they have accepted because they wanted to take the credit that yeast is used in bakery, yeast is used in alcohol production, ethanol production, and yeast is industrially important. Yeast is important in the case of genetics and so on and so forth. But it is eukaryote, and all the microbes are prokaryotic. Still, they wanted to include yeast in their as their patent organism. So neither plant scientists nor animal scientists nor microbiologists accepted. Therefore, fungi thought that let ourselves be a kingdom, and therefore it has become a major kingdom, mycota. And we are, again, it got segregated into different kingdoms. Eumycota, it has got Basidiomycota, Ascomycota, Zygomycota, Chytridiomycota. Earlier, there used to be a Deuteromycota, fungi imperfecti, but now it has been removed because of the fact, for example, penicillium you take. Penicillium is the conidial state, whereas its perfect stage is Talaromyces. And if you isolate ascospores and put it on agar medium, it has to produce penicillium state. That they have proved in certain cases, not in all penicillia. There are 2000 penicillia, and all the 2000 penicillia will not produce perfect stages. Like that aspergillus, the perfect stage is Euroseum. It's clistothecial. It doesn't mean that all aspergilli will produce perfect stages. Only Emersella nidulans could produce the clistothecia. So like that, the scientists have come to a conclusion that there must be perfect stage for each fungus, and only such fungi should be considered, because the occurrence of conidial stage is only a momentary, a temporary phase, and its perfect stage may be lying in the nature elsewhere. But unfortunately, mycologists and brainy minds could not find what are the perfect stages of remaining fungi. We have got only few teleomorphs, that is perfect stages of only few fungi. 30,000 Ascomycetes are there. So then next comes Heteroconta, that is under Chromista, where you can find, it is called Pseudomycotina because of the fact that Oomycetes like Saprolegnia and Pythium Phytophthora, they come under Oomycetes. It has got two flagella. That is the reason why it is considered under Heterocontae. And then Hypochytridiomycetes. Hypochytridiomycetes are very few genera and few fungi are reported from world, but not from India. Only one species is reported. Nobody has worked on that. I told you that whichever is easy, they will work out. Whichever is not easy, they will leave it for others. So coming to the others, Myxomycetes and Plasmodiophoramycetes, earlier they were included under fungi, but now they have got separate phylums under the kingdom Protozoa. So this is a very simplest classification. Now, what is fungal taxonomy? The fungal taxonomy deals with collection and then isolation, subculturing, single spore isolation, colony formation, morphological studies, vegetative structures, study of reproductive structures, microscopic and macroscopic characters in the case of higher fungi, and then ultramicroscopic characters, the cell wall composition and internal contents, and then chemical profiling of secondary metabolites, because each fungus has got its own metabolite. So after growing them in culture, what is that secondary metabolite that is secreted by this fungus? Scanning electron microscope studies, because beautifully ornamented ascospores are there, beautifully ornamented basidospores are there, and then computer assisted keysite to be developed. I think this is later part has not been worked out in India. Still, we are only looking into the manuals and monographs. And coming to the molecular taxonomy, immunological methods, they are very costly, and therefore very few people where it is important they are using. Enzyme pattern, Dr. Pitt has isolated, identified number of Pythium species and Phytophthora based upon pectinase enzyme. Carbohydrate pattern and protein profiles, and guanine and cytosine molecular ratios, DNA hybridization, nucleic acid probes, and rRNA sequences. All of you know RFLP, PCR, RAPD. Now DNA barcoding also has come. Now carbohydrates, and it related to fungal taxonomy. Carbon is the source for the growth of the fungus. Monosers are useful in yeast taxonomy. You can, based upon polyols, glycerol, arbitol, mannitol, you can identify many fungi, and then you can, like chitin and beta glucanase, all this composition will help us in the case of characterization of fungi and identification, classification. So they are all useful in the case of fungal taxonomy. Now, secondary metabolites, they are reliable and diagnostic taxonomic characters. Of course, we have morphological characters, but secondary metabolite data have yielded clearly species-specific data, and a large number of isolates we had to screen and from different geographic regions, and then occurring on different habitats, those fungi had to be taken. Like, for example, Aspergillus, Fusarium, Penicillium, and all of them have been identified based upon secondary metabolites. You take Penicillium griseofulvum, polyketides, griseofulvin, it yields. And based upon this griseofulvin content, you can say how many strains of griseofulvum are there. Terpenes and like Taxol, Taxomyces andriana, this is the fungus which is present in Taxus brevifolia. And it has yielded Taxol, an anti-cancer drug. It has become first multi-million-dollar drug in United States. That is the first drug that was created, and then multi-billion-dollar worth of drug. It is from that of the fungus Taxomyces. So we have different types of these secondary metabolites which will help us. Now you can see these are the biodiversity status. This is left is Alomyces, and this is the Pythium, and the next one is lower one is Saprolegnia, and that is the oospore of Eubonia and oospore and Anthridia of Phytophthora. You can see Amphiginous Anthridia at the base, and see there is a carotenoid pigment in the case of Alomyces, upper one. You know that carotenoid pigment has become very important in cardiotherapy. See, when you look into the fungus, you look at its importance also, how best it can be used in agriculture, how it can be useful in pharmaceuticals, how it can be useful in waste recycling, how it can yield enzyme. All these things have to be worked out. Simply working nowadays only on the taxonomy and molecular characterization only is not important for us. You know, Emerson, I spent 11 years in understanding the life cycle of 6 species of Alomyces. And Pythium releases the zoospores into a vesicle, beautifully ornamented. You can see nobody has asked Pythium to release the zoospores into vesicle. It is a life form. It is a life form. It has got its own way. Why somebody always twinkling his eyes? It is a habit for him. We don't know why all the people are not doing that. So it is a life form, and we cannot change certain habits, and we cannot change the morphologically inventorized or whatever that is established by the fungus. If you want to that Pythium should become Saprolegnia, some people are working only on the basis of genetics, but no, they could not succeed in that. Now, in Saprolegnia, there is diplanetism. That is, 2 motile stages are there, and sporangia proliferation is there. So this is very important, and all this is why I'm telling, because when you explain to the students, they should get attracted toward the subject. And until and unless you give them some important data and the attached information, they will not get interested. Nowadays, somebody sent a PDF file to me that botany is declining and people are not interested in botany. But I tell you, earth formation is because of microbes. And you know, whatever the life is there that is established on this globe is because of the fungi, microbes, and even plants' existence and human existence. What you are there on the soil standing is because of only the microbes and fungi. The soil formation is because of that. So you just cannot say like that. So these things are very important to look at. And then these are all some of the, you go to any waterfall, and then take the foam sample and observe the fungi. You can find all these fungi. It indicates they are entirely different in their shape. In order to adjust themselves with the water currents, the shape has been defined. So these fungi are very important in the energy, making the aquatic ecosystem energy rich, and also they are the bioremediation, they are useful. They are also clean the environment by means of, that is, they will remove the heavy metals and pollutants, and they are also indicators of non-polluted waters. So these are some of the fungi which are there. And see, now coming to the mucorales and zygomycota, and these they produce by means of sporangiospores, that is eplanospores, non-motile spores inside the sporangia. One columella is also there. In Rhizophus, rhizoids are present, and then it produces sexually heterothylism black sea. 1904 has recognized this phenomena. See, 1904 way back, and that is still, it is valid investigation. And neurospora has been worked out by Tatum and Beadle, one gene and one enzyme theory. Still it is valid. So we have such kind of scientific data. And phototropism in the case of Phycomyces, Phyllobolus, hat fungus. The phototropism was discovered in the case of fungi for the first time using Phyllobolus. You keep the dung pellet nearby the window and sprinkle some water, the Phyllobolus will grow and it will bend toward the light. That is the phototropism. So we have such kind of fungi where biochemistry, food industry is dependent on mucorales, and a lot of industrial products are there. And many of the fermented products which are sold in the European countries and in US, UK and USA, they're all nothing but fermented foods. Aspergillus oryzae, you take, that is used in the fermentation. Yeast is used in the fermentation. Many fungi are, Angkok in the case of China, the red color is because of the fungus. They like the red color, therefore they put the fungus inside the rice, and that will give a color, a product that is comes out of that particular one. So we have lot of industries which are dependent on the zygomycotina. Around 10,000 fungal, 1,000 fungal species are there. But you see, don't think that all the fungi are very good and they are helpful to the human welfare. But some fungi like us, we don't have any chlorophyll, they don't have any chlorophyll. Therefore, the Succinia wasformis is also one fungus which is parasitic. It is a secondary parasite. In many of the soldiers who got wounded during the what we call as war in recent times also, it has become that an important secondary parasite, and they have to lose their limbs and hands, and because of the secondary infection. And we do have, in the case of pandemic, in the viruses and where the people are diabetic and all that, inside the lungs also, there was a mucormycosis disease was noticed in the case of corona affected patients. And it is not the will of the fungus to go and enter, but you might have moved in certain areas where the fungus has been floating, and that is the reason why it has become very important, and then it has entered the nostrils. Maybe house dust and other areas are also maybe there. Now these are some fungi which are many fungi like Cutomium, Ascomycotina. Cutomium produces tons and tons of cellulose, which is useful in textile industry and also in the garment industry to make the stones, diamonds to shine. And they are dependent upon this particular fungus, cellulose enzyme. And then you can see Ophianectria, that is the bioluminescent ascospores are there, which are very useful. In other words, that is bioluminescent phenomena is an important method. And 2,840 research papers are produced on this particular fungus, Neurospora crassa. And 6 Nobel Prizes have been awarded for this particular fungus where has worked. And academic that is publisher from USA, they have published around 6 volumes. And then Pezaiza has been grown on the first time we have grown on the agar medium and shown that it is also a very good decomposer. And Basidiomycotina, I need not tell you, the multi-billion-dollar industry of mushrooms that is existing in the Europe is because of Basidiomycota. The agaricales and members of Agaricus campestris and all other edible fungi, pleurotus, and as well as Valvariella valvaceae. And this group comprises largely fleshy fungi, and we have got 10,000 fleshy fungi that are there. 80 fungi have been identified as edible, and but only 3 have become culturable. That is, button mushroom is culturable, and paddy straw mushroom is culturable, and pleurotus has been culturable. But that is Agaricus Bitrochis is culturable. In Bengaluru, horticulture department has done it, and some other fungi also they are making efforts and to culture them and bring them to the commercial side. And Flammularia is in the case of China and other countries. You see, China, Thailand, and Brazil, and all these countries are #1 in the mushroom technology, and we are lagging behind. And Eastern countries are doing very well, and we have got 10,000 species, but we are not able to generate any kind of economy and help the country. That is one. Of course, it has become household industry in Maharashtra. It has become household industry in some other places. Now coming to the Marcella esculenta that I was telling about, this under Aplarcars it grows, and then these are the button mushrooms and all that. And you see, you can see the coprinus, which is there are, you see, 120 dyes which are produced by the fungi, and this produces the black dye along with the plant dye. It is also used in the elections to put dart on the finger. And Schizophyllum commune, it yields anti-cancer drug, the lower one. This is graphium, broomstick-like structure, which we have isolated from. It causes onychomycosis, eye disease, and from the eye infections of L.V. Prasad Eye Institute. And there are 20,000 fungal species in the case of fungi imperfecti, which is a large group. This is one fungus, and we have discovered 0.01% of sodium chloride can cure this particular disease. Like that, Aspergillus produces 7 enzymes and many organic acid and inorganic acids. It is an example for the parasexuality, metatic recombination. For the first time, this has been discovered in this particular fungus. And also growth promoting substances are secreted by Carvularia and other fungi. And now the recent group that has come into existence is endophytic fungi. This is a very important group. It doesn't cause any disease. They are there inside the tissues, and this tissue is colonized by the fungi. These fungi elaborate a number of metabolites, secondary metabolites, which have become industrially important, pharmaceutically important. And we had one fungus, Pestalozia, which we have made a DBT patent. We have submitted, and it is yielding an anti-cancer drug also. So we have such kind of studies, and this is a recent origin that people are working on endophytic fungi. This is about keratinophilic fungi, which cause, they are present on the human skin, they are present on the nails, feathers, hooves, and horns of the animals. And see, Chrysosporium, Arthroderma, Epidermophyton, Malabranchia, Microsporium, and then Trichophyton. These are some of the fungi which are present, and Chrysosporium tropicum could be considerably high in the case of many areas. And also, as I told you, Candida auris has become an important fungus in the case of hospitals. A lot of publications have come on the human diseases and also on the hospital fungi, allergenic fungi, and so on and so forth. And Dr. Alim Khan has worked on aeroallergenic fungi. Very important contribution he has made from Nanded and other areas, and he has published that data in an international journal, and very good data he has submitted, which is very useful. And glimpses of the fungi is 1904, Blacksley, heterothallism stimulated studies on sexual control, and Barjeff, Neep, and Inlergen laid the foundation for fungal genetics. Fleming has discovered penicillin, and here what happened is a little bit story is that Fleming is a scientist, no doubt, medical doctor, but he was working on bacteria and fungi also. And then his wife was working as a technical assistant. And when she was looking into the petri dishes and washing them, she could find that one fungus, blue fungus, was controlling a bacterial colony. So just that data she has shown to Fleming, and Fleming immediately jumped onto that particular fungus and came into that. So biochemical genetics and mid-90s, the favored eukaryotic molecular biology. This is $16 billion worth of fungal biotechnology is in bakery, brewing, cheese making, mushroom cultivation, oriental fermentation, single cell protein, alkaloids, antibiotics, enzymes, gibberellins, and then cyclosporin. Without that is organ transplantation is brought revolution because of cyclosporin, organic acid, polysaccharides, vitamins, paper industry, biological control, and bioremediation of polluted soils, coal solubilization, ultra coal production, and dye industry, microencapsulation, biofertilizer technology, waste recycling. Everything is dependent on fungi and bacteria. This is taxa, this is penicillin, and you can see on the other side, Dr. Fleming, he has this ergot alkaloids, and it is very useful medicinally, but during the childbirth and all that, and it is also useful in some other forms also. But if you take in larger quantities, you will be elsewhere, flying in the air, and you will not be on the ground. And ergot alkaloids are very important. 7 alkaloids are there. Metabolites, beta lactams are very important. They are antibacterial, and they combat a number of bacterial infections, and beta lactams are 50% of world market is dependent upon these beta lactams produced by the fungi, griseopolvin, puromethylene, physitic acid, sardarin, and all that. These are all some of the insecticidal and nematicidal metabolites which are secreted by many fungi, and many pests can be controlled by means of this Beauveria bassiana controls coffee berry borer. Metarhizium is useful in controlling mosquito larvae, and like that we have got a number of fungi which are useful. And then these are all some of the antibiotics which are useful in industry. You can see the industry has produced a lot of, you can look into this particular one. And then metabolites, they are produced riboflavin, which is commonly used, citric acid, and then kojic acid, itaconic acid, alkaloids, gibberellin, growth promoter, beta carotenes, and physitic acid. And bioabsorption, that is biofertilizer, Biosol is from that of Penicillium chrysogenum. It is commercialized, and minerals are solubilized by Aspergillus niger, very common fungus. Heavy metal pollution can be controlled by means of mycorrhiza and Aspergillus. These are some of the commercial enzymes which are available in the market. And see, to make the capsules coated with many of the cellulase and other things, and also as a products, and asparginase, amylase, catalase, cellulase, and then beta-glucanase, glucoamylase, glucose oxidase, hemicellulase, lactase, lipase, pectinase, protease, rennet, tannase, gyalinase. All these are used in commercially. They are available. A lot of marketing is there. And fungi are used as a biocontrol agent. Aspergillus, Beauveria, Trichoderma, and Trichoderma used to control many of the soil-borne and root-borne diseases. PGPR is a growth promoter. And we have got a lot of fungi also which are growth promoters. And these are also only, they control a lot of mosquitoes. And because in India we have got mosquito problem, therefore I am showing this particular slide. Unfortunately, Coelomyces is available. 1915, Iyengar from Madras, he has isolated this particular fungus, but its sexual stage is occurring in Africa, and they could not mate the asexual and sexual mate together. They should not bring them together. That is the reason why still the life cycle is incomplete and mosquito problem is still remaining. And mosquitoes also have become very virulent. And because of the spray of many of the chemicals to kill them, they have become very virulent, and Coelomyces is not able to, metarhizium also is not able to colonize, not able to kill the mosquito larvae also. So it is our own deeds which will bring the fruits, and it is foolish to think that somebody will come to the rescue when you are in crisis. And these are cheese and meat, yeast and mycelial fungi, recombinant DNA technology. All these things are very important ones. And then you see Asian fungal, see Sufu and to the Ragi, Mura, and these are all used, and these are fermented products which are sold in the, especially in the Europe and the United States and UK. These are the fungi which are used in the food industry. And such kind of fungi are also available in India, but we are not coming forward to I told you that Angkor Monoascus purpurea is the red pigment fungus that is used by the Chinese. And soya sauce, which is on our table, is because of Tarlopsis and Zygosaccharomyces, which are the common fungi occurring. And the next 10 years, no product of modern biotechnology will be equal to that of the alcohol. And alcohol is controlling a great trade, hormones, antibodies, and other pharmaceuticals. And the following directions are to be seen. Immobilized biocatalysts, genetically modification of raw materials is important, and modified yeast has to be. Yeast has not been studied in India, unfortunately. Not much work has been done, and we don't have any culture deposition of yeast also, and very few cultures are available. But in sea alone, you'll find 200 yeasts, which are industrially important. Fungal carotenoids, beta-lactams, catecholamines. See, market sale in the United States is 500 million. And we have such kind of fungi which release this chytromyces and black sea and other such kind of. Mycogrow is a biofertilizer. It is a preparation of mycorrhizal fungi, promotes faster growth, speeds up the transplant recovery, and reduces the need for fertilizers. And then these are all some of the tablets which are available in United States and also in India. It contains 150 tablets, costs around $30, ectomycorrhiza and also endomycorrhiza, and that is am fungi. And these are the one tablet for one plant that is good enough. And these are all commercialized fungi which will control the weeds and insects, Quran, and then divine, coligo, cast, mycar, mycogrow, that is mycorrhizal fungi. Bina tea is a trichoderma product, biocontrol agent, and then commercialized fungi, biofertilizer, Bysol, baker's yeast is sold in the market, soy sauce is sold in the market, beer, wine, alcohol, it is because of yeast. And fungi is traditional medicine. Ophiocordyceps sinensis, 7 species are there in Nepal and India, and it infects moth and damages patches. It yields stress-relieving tonic. Chinese people could get gold medals because of this particular tonic, and it might be anti-cancerous. Caterpillar fungi are used as gifts. A large gift box is about $400, and tonic is a growth promoter and immunomodulator. Therefore, it costs around $27.53. And it is anti-cancerous also. Therefore, let us look into this particular fungus which is available, but it could not be grown on agar media. And this is Ganoderma, is used in cosmetics. It is anti-cancerous used. And we have got 7 species in India, but all the 7 species have been taken by Malaysia, and they are producing the tablets and they are selling to us. And then this is the recent one, Lesiodiplodin from the top of New Zealand. It is an antioxidant, and cyclosporin, which has brought a revolution in organ transplantation. And it is 90% of plants are mutually beneficial because of the mycorrhiza and other fungi. So the earth is dependent on fungi, microbes, and fungi. So these are all symbiotic organisms. I don't want to go into details. Arbuscular mycorrhizal fungi. You can see on the right side, we have inoculated arbuscular mycorrhizal fungus, glamus, and then fasciculatum on the left side without any glamus. You can see the productivity and then healthy plants that are produced. And this is new species which we have named. As I told you, we have 120 new species. This is one species, Glamus hyderabadiensis, and Beauveria bassiana we have manufactured, and we are selling it to the farmers to control some of the insect pests. And you can see this trichoderma also, and then like that. These are some of the diseases, very important ones. This is Ajanta paintings. We have lost around 7,000 crores worth of paintings, and because of the infections caused by bacteria and fungi. It is not the mistake of the bacteria or the fungi. It is the people who carry with them the eatables and the shoes, along with the shoes and other things. They enter into these sculptures and paintings where they are available, and they are seeing, and they eat and all that. And what happens is that on that particular debris and the dust, these fungi, bacteria, and other things will be growing, and they blow onto these. And when there is moisture inside the caves, and you will find that they grow and multiply, and they will be damaging this. Later, late blight of potato, which has made the Ireland people to suffer, and it was become epidemic, and there was economy loss. And then Ceylon's people have forgotten coffee drink, because of the fact that coffee rust Emilia aesthetics has made them to forget coffee. If you don't control the plant diseases naturally, we have to go for hunger and poverty is the next result. The economy will be lost. And healthy seed is important on the right side. And if it is contaminated and infected by fungi and bacteria, I think our agriculture will be lost. And you can see, anyhow, nowadays everybody is looking at the Google Mata, and therefore fungi want to read many of the textbooks and which are there in the library. They are, because of the humidity and all that, fungi are growing on the, you can see black, Aspergillus niger, which is growing, and Carolaria, which is growing, Cladosporium, which is growing on biological abstracts from that of the library I got. And people should use all the textbooks and they should go into the details of ancient textbooks. Unfortunately, and this is happening and everybody is dependent on Google. Always Google doesn't give you the original data. Sometimes you have to depend upon on our own data also. And you can see neglected groups of fungi, chytrids and hypophytrids, trichomycetes, levalbinomycetes, lapilomycetes, yeast, waterborne colony, gastromycetes, sooty-moles, nematophagous fungi, marine fungi, extremophilic fungi, crop-wise pathogen fungi, animal pathogen fungi, medicinal mushrooms. And in my honor, four fungi have been named. And of course, I don't want to take all those names. And fungal taxonomy should not become forgotten science. And challenges and solution, it is there from that of the fungi and microbes to the human mind. And then new discoveries. This is what we have discovered: 20 new genera, 120 new species, 500 new actions, and 2 new fungal genera and 3 new fungal species have been named in my honor. And around 1,500 cultures we have deposited in the national and international culture centers. We have brought monographs on soil fungi and trichoderma, fungi and aquatic habitats and aquatic fungi from Andhra Pradesh and Telangana. We have published 640 research papers now. It is 640 and 220 are related to taxonomy. And God made the species, but man made genera. These are some of the discoveries we have made. And these are some of the Badradale and other things, some new fungi we have described. And then fungi in synthetic biology, they have become very important in industrial biotechnology, in proteomics, genomics, and metabolomics, omics sciences. And then, there is one Amanita varna, which produces its mycelium up to 26 kilometers. And this is what that has been reported, false stem in 2005. And micro nanotechnology has come. Already you heard that particular lecture, and they are also useful in the disease control. And then biofuel fungi, many fungi are useful in biofuel production. Fantastic fungi, mushrooms are fantastic. And Subramanian said, the most recent examples of harnessing fungi power for human needs highlight the massive potential and future possibilities of this vital area. No fungi, no future. So these are all some of the scientists who have worked on fungi. My teacher, Professor Ramarao, first person. Second, Professor John Webster, with whom I worked on PDF in England for 1 year. And then G.P. Agarwal, who has worked on keratinophilic fungi. Late C.V. Subramaniam, who has contributed 32% of Indian fungi. Thirumalachar, who has discovered antibiotics from India, laid foundation. B.S. Maharatra worked on, from Allahabad, worked on mucorales. K.C. Mehta has worked for about 22 years on rust fungi, especially on wheat rust. And Sadasivan has worked on enzyme theory and toxins. And Mundkar laid the foundation for the present IARI, Indian Agriculture Research Institute. And this is the university which has given me bread and butter. I'm thankful to my teachers, my parents, my family, and my research scholars, and all my students, and to the university authorities, and to every one of you. And thanks to Dr. Alim Khan, and to all specialists and all scientists of the country. I dedicate this to all those people. Thank you very much.
सर शेयर स्क्रीन बंद करिए सर.
Share screen बंद करना. I will do it. Just I will have some water and then come. Hey.
Good morning, Rao sir.
Good morning.
Yeah,
Sir,
coming.
वीडियो ऑन कीजिए सर,
Coming.
वीडियो ऑन कीजिए, एक फोटो लेंगे.
Yeah.
Please, participants, all of you, once you can start your video, all of you.
Hello. Yeah, tell me, sir.
Please quickly, because our next speaker is ready. Please, all of you, quick. Deshmukh madam and others also, all I have requested already in the WhatsApp and mail and everywhere I have requested. Please, still 2 lines are without photos. Dr. Jalandhar, Dr. Sunil Kumar, Parvati ma'am, Ranjani ma'am, Suvarna ma'am, Kalyani ma'am, Shipra ma'am, everybody, please quickly. Dr. Srinivas, you too. Today is last day, please, quickly. Please, we are waiting for photo, please. सर रुकिए सर, आप चारी सर रुकिए, एक फोटो हो जाने दीजिए. Dr. Mahendra, you too can start your video. Dr. Mahendra, please, we'll have one snap. Dr. Srinivas, don't delay. See, I have requested already yesterday. Please, we are waiting for one more row down. All others also, rest of them also. Dr. Mahendra, you are in meeting, it seems. Please, you quickly switch on your video. Rare opportunity. We have so many people from different places.
Yeah.
We have got from Karnataka, Maharashtra, Telangana, Andhra Pradesh, and so big assembly. I think it is. Yes. Dr. Mahendra, our zoology. Yeah, you also. Yes. 2 more people are remaining now. Swarnalata madam, please. Only 2 people are left. Please quickly. And where are you, Deshmukh ma'am? You are not visible. Yeah, okay. Right, smile please, all of you. Done. Chaliye.
So, Dr. Alim Khan,
Sir.
I would like to say 2 sentences or 3 sentences. First of all, I would like to thank Professor Dr. R.R. Rao.
सर वैलिडिटी में बात करिए ना, वैलिडिटी में बात करिए, वैलिडिटी प्लान करेंगे.
वो कब है?
लेक्चर के बाद है सर.
After 4 o'clock?
Yeah, after 4 o'clock for 5-10 minutes.
But first of all, let me thank Dr. R.R. Rao, our honorable sir, and who has contributed a lot for this Indian ethnobotany and plant taxonomy. He is a rare species, endangered species in our country, and through him, many scientists could mold their lives for this particular country, who have become very good specialists and directors, and they are in holding different positions. And he is a person with a lot of magnanimity, and he is a person with all such kind of qualities to help others. And we are very fortunate to have Dr. R.R. Rao, who is known for his benevolence, who is known for his kindness, who is known for his knowledge. And with these few words, I request Dr. Anvipan to introduce our Honorable Dr. R.R. Rao, sir.
Oh, thank you, Dr. Manoharachari, for so nice words about me. I am really honored to hear nice words. Thank you so much. And you are equally, you are equally a rare species. equally a rare species. Well done, commendable work. Almost
Yeah,
500,
yeah.
600 papers you
On
have put.
one platform,
Equally great in your area.
we
Yeah,
have
thank you,
legends
sir.
today on
Yes.
one platform from two different areas. Thank you so much, sir. You can start sharing your screen, sir. Rao, sir.
No, sir. What you want? Conservation today, and what is the
Yes,
other one you
yes.
wanted?
Medicinal plants वाला. दोपहर में हम
Medicinal plants and conservation.
systematic दोपहर में करेंगे.
So should I go for conservation now?
Yes.
Okay. So share कर दो इसको.
Thank you, participants. Again, we'll have one more snap for after the lecture. Thank you.
Okay. Yeah. Thanks, Dr. Alim Khan, Professor Manoharachari. This is indeed a great opportunity for me to share some of my thoughts on the conservation of threatened habitats and the threatened plants in our country. What we have done, what are the responses that India has shown towards conserving our biological diversity. These are the few things I will try to discuss in the course of my, say, one and a half, 1 hour, 15 minutes. I think I will try to do justice to this subject. Again, please understand, threatened habitats. Yesterday also I told one example, I told sacred groves, for example, threatened habitats. Habitats are the ones which support the diversity. There are many such habitats which have become critically endangered in our country. Not only the habitats are disappearing, but the associated species also disappearing. So therefore, restoration of threatened habitats is very important. For doing so, we should know which are all the real threatened habitats in our country. Threatened plants, yes, we know a large number of, I'm going through my talk, but then let us see which are some of the threatened habitats in our country, right? Now, Indian region, yesterday I told, is one of the superlative in all respects. It's one of the wonderful areas. Biodiversity is very rich. And I told arbors, almost 78% of the world's all recorded species we get here. Of the 36 global hotspots, 4 are present in our country. I told this. And one of the 12 centers of origin of diversity of cultivated plants, according to Wevilov, Hindustani Center of Origin of Cultivated Plants, that is another one. Roughly 8,500 vascular plants are recorded, of which roughly 500 endemics are there. 5,000 endemics are there in our country. And these endemics are again scattered in 4 different regions: East Himalaya, Western Ghats, and Andaman and Nicobar Islands, different areas. And there are 30 large families with more than 100 species. That is the characteristic of our country. And cradle of flowering plants, I told yesterday. I think this you should always remember, because of the existence of large number of primitive flowering plants in our country. Our region is considered to be a cradle of flowering plants where angiosperms have originated and diversified. So these are all the unique characters that we have. When you talk of mega diversity centers or mega diverse countries in the world, all the green ones which I have shown, all these 14 countries, which are all mega diverse countries. India is very fortunately, it's one of the mega diverse countries that we have. And adjacent to us, which is a very strong competitor for us in all respects, is China. So please remember, anything now we do, in fact, we do much better than what Chinese are doing. But unfortunately, we are lagging. I will tell you where we are lagging. But anyway, I think the expertise in our country is far better than Chinese. So therefore, I have every hope that the future younger generation will take it up. This is just only yesterday I showed, and please again remember that there is enormous amount of species in our country, 18,259 species. As I said, there is no sanctity to this. It may go on changing every time. And our expert, Professor Manoharachari, is also there. Fungi, we have written in the world 75,000, and in India 15,500 species roughly recorded. Perhaps he may correct this, or he may say more or less something else, and they can see this. And This only gives the strength of the biodiversity that we have got, and which also shows how many endemic plants are there. Endemic plants are very important for us because it is the endemic plants we can always claim it is ours. Even if somebody takes our genetic resources and develops a product in some other place, we can always sue him in the International Court of Law and get the entire royalty to our country. So that's the advantage after the CBD, after the CBD has come. So these are all the endemic plants that we have got in our country. And one of the reasons yesterday I told is the all the habitats that you can think of in this world, you find in India. Any habitat you can think of. That's one plus point for us. So therefore, our diversity is enormous in our country. And this is divided in, scattered rather in different biogeographic zones like this. Different colors I have shown in different. There are 12 biogeographic zones in our own country. Depending upon the authors, I think it also changes. Many people have made it into 8 biogeographic regions. Some have made 24, 23. Some have made 12, and some have made even 11. So according to me, in my own book, I have made it 12 biogeographic zones in our country. Now, let us think of some beautiful plants. And I put this question, can we afford to lose these resources, such beautiful resources? Can we afford? Now, the question is whether it is useful or not will come later on. But think, such beautiful plants are there. Such beautiful plants are there. Can we afford this? Can we miss these plants? This is the question now. Can we afford these plants? How many of them are medicinal? How many of them are, biodiesel plants? How many? All these things that utility will come later on. Such beautiful plants, can we afford? See, Ceropegias, you imagine the enormous variation, diversity of Ceropegias. Now, this is the unique country that we have so much of diversity and all of them. And then our economically important Myristica, for example, Myristica is Jaikai, or also it is called the Yaril, which is having high value. It is medicinal as well as a spice. It is used, extremely costly. Also in the market, it is sold. Myristica dactyloides, Myristica malabarica, some of these Myristica nematoneata, these are all the genera, very highly economically important ones, but scattered in the forests, and many of them have not come under cultivation. Can we afford to lose all this? With the yesterday I told Western Ghat has become the hottest hotspot. Hottest hotspots are those areas where 90% of the original forest has been destroyed. Now, can you imagine now if this kind of forest plants are completely disappeared from our country? Even tomorrow, later on, if you find that this is a wonder plant, this is a Sanjeevani plant for us, we cannot bring it back. So therefore, the question is, can we afford to lose these things? So we cannot lose. Biologically curious plants are quite numerous, so beautiful also, and then so numerous. This is Utricularia in the Khas Plateau. This slide also yesterday I shown wonderful impatiens lai. So ocean of flowers, and the utility I talked about. Now, suppose you destroy this habitat. Habitat is, I told, in the flat topped the hill mountain tops in the lateritic soil is the real habitat for this. Now, can we destroy this kind of habitats and lose? Aquatic systems, you can imagine in the aquatic system, so beautiful plant. This is Eriocolan, very beautiful plants, and you can always use them in the man-made settings also. You can use those things. Now, we cannot afford to lose. One introduction of one aquatic weed is sufficient enough to remove all these plants. I think we cannot. This is, of course, having a phytogeographic significance. Recently, Yadav has reported this, Trichuria concanensis. It has got very unique position in the phylogeny, particularly when you discuss, say, APG system of classification. I think this throws some light on that, whether it is a monocot, whether it is a primitive dicot, this kind of discussion we can have. So many numerous orchids are also there, very beautiful. On this, the economy of the country can be, changed. So beautiful orchids are there. Can we change these things? Can we afford to lose? We cannot afford to lose. Rhododendrons are there. Can we afford? No. Beautiful rhododendrons we cannot. I told about the divine ring soma. Professor Manoharachari is there. I think you can see this. It's a beautiful Amanita muscaria. Now we still do not know what is the real surrogate of soma. Suppose we have been able to resurrect soma, economy of the country can change. We can break down the monopoly of Scotch whiskey. I think all that possibilities are there. This is Ephedra gerardiana is said to be one of the very strong surrogates, whereas the sarcostema is a flowering plant. This is also occurring even in Telangana region also, you will get this. This is also said to be a very strong surrogate of Soma. But now most of the people, including Swedish botanists, thinks that the Amanita muscaria must be the real Soma, which was in usage some 5,000 years ago in the Vedic literature. Also, in this Ashtanga Hridaya says, all plants have potential medicinal value. There is nothing in this universe which is non-medicinal. That means to simply say all plants are medicinal. Some we know, some uses of the other plants we do not know. So therefore we say out of 18,000, 8,000 species are medicinal. What about others? They are also medicinal. But as he says, every plant is useful, but some virtues are known, some virtues are not known. Now, when this much of diversity is there, uniqueness is there, side by side, there are many threats operating on this biodiversity. This is number one. Yesterday, I talked very briefly. Now I will try go a little elaborately because Dr. Hamidullah Alim Khan wanted that I should talk conservation. Now, human population particularly is threatening the biodiversity. It's all anthropogenic disturbance only, most of this. And even the invasion of alien species in the country is also from human. I don't think there is anything natural. Very rarely, sometimes we get by way of tsunami or earthquake and all that. Otherwise, most of the threats that is being operated are all anthropogenic in nature. Now, increasing human population, for example, I just say here, by the year 2110, global population is likely to have expanded to 10.5 billion or 2.5 times as many as the Earth is now supporting. You can imagine, by 2.5 times more. And of this population, of the 10.5 billion, 9.1 billion will be living in developing world, where biodiversity is also rich. So in these areas where biodiversity is very rich, human population is also expanding like anything. Out of 10.5 billion, 9.1 will be in the developing countries. So therefore, you can imagine what could be the impact on the biodiversity loss, on the biodiversity. Naturally, with expansion of human population, all the biodiversity is going to be lost. So this is another one. In India, these are all the major factors that is being operated now. Why the plants are, or the habitats are shrinking is because of this. Forest clearance for extension of townships. When human population increases, naturally, we have to go and cut off the forest and build townships. That's one. Selective removal of certain groups, say medicinal plants, ornamental plants. Some of these medicine groups are selective removal. They are all collected for their medicinal use or whatever it is. That is another thing. I told yesterday an example of aconitum. Aconitum in the Himalaya, for example, very costly, almost 4,000 rupees the tubers cost, and the local people go collect it. And when they collect it, what they do, if the tubers are immature, they will immediately throw that. They will not take the trouble of again digging and planting it again. So unscientific harvest, selective removal, all these things are causing threat to the medicinal plant group. And draining water from swamps. Now, swamp is a unique habitat in our country. It is a very unique system that we don't find. In fact, swamp forests were said to be once a common swamp starting from the Western Himalaya to the Eastern Himalaya. It was a continuous stretch of swamp. But today, in all the Indo-Gangetic plain in those areas, the agriculture fields have come up with the reason only one patch in the Dehradun Valley. In the Doon Valley, we have one swamp forest, and in the extreme northeast, we have one in the form of the what is that Kaziranga National Park. That is the one. Only two places we have left with swamp forest. Spread of forest fire, I think you know this. It's a global phenomena everywhere. Spread of forest fires. That is another one. And shifting agriculture. Yesterday I told, I will not repeat this. Shifting agriculture in northeast region. There, it is said in the northeastern region is again a hottest hotspot. Understand this. It is said there is not a single area which has not been zoomed at one time or the other in the past. I think this is the sweeping. Actually, there is not today in the entire Northeast, except for the sacred forest, in the entire Northeast, there is not a single area that has not been left at one time or other to be zoomed. I think it has been zoomed. So mining activities, these are some of the factors. I think I have shown some of these yesterday also, so I will skip. The invasive alien weeds, again, that is another percent. 40% of the Indian flora is introduced or naturalized. If you see the Indian flora, 18,000 species, out of which 40% are introduced and are naturalized. Now, forest problem is not there with naturalized aliens. Now, the alien flora, many of the plants are naturalized in our country, almost 40%. Naturalized species are those which are living in harmony with the native flora. So there is no problem of naturalized flora, but it is only the alien adventive weeds which have come recently and have eliminated our completely local flora. And these are all exotic weeds which are all dangerous to our flora. So such flora, there are large number of, I think, more than 100 to 150 such invasive weeds in our country, in different parts of the country, including aquatic ecosystems. Also, these species are there. Now, this is composite Artemisia. This is again yesterday I told this Hypomia carnea. I think in your region also, in most of the region, it is very common today. And once it enters the area, the entire area is covered. It will not allow any other plant to come up. This is cassia, this is mucuna, some of these plants that have come. In the aquatic system, if you see, this is Pistia, for example, this is the plant, whole area covered. The photosynthetic system completely cut off. If there are any species growing, it will completely cover the photosynthetic surface, and the entire plants, they die in the aquatic system. So this is a major threat that is happening in our country with regard to aquatic areas. I told about this Brahma Kamal yesterday also. Again, this is for the local people collect the selected plants only they will go, and that is what I always call selective removal. I told about this case. This is Brahma Kamal, one of the important medicinal plants, highly fragrant, highly aromatic plant. This is an inflorescence. Once the inflorescence is removed, there is no way for the plants to multiply. So this habitat has shrunk so much. Today, the Uttarakhand government today has banned collection of this plant, and stringent rules are put in force now. Now people cannot go and collect this plant. Now there are also in the northeast, some of the areas, is a Catamyxis beccariides, which grows in steep cliffs like this, very steep. It is an endemic plant, but highly important medicinal plant. But at the same time, local people collect this plant. Can you imagine this? He has one ladder above the other, he has attached, and he is going and collecting plants. So that is the selective removal of these plants. Now here is a plant. This is Erimostachys superba. You can see here highway, Dehradun Delhi highway passing through. A very beautiful plant is standing just near the highway. Now, do you think it will not attract the travelers? Many people who come there seeing that so beautiful plant, they want to take this, they will immediately lift this plant without knowing that it has a tuber, very strong tuber down below undergrowth. So therefore, they only take the aerial portion. With the result, the reproductive part is removed, the plant is not multiplying. Today, there are hardly 27 or 28 plants in the particular area, that is Mohand. That's the exact place is Mohand on Dehradun Delhi highway. And there are only few plants, and the existence of these plants for further is only a matter of time. Look at this, same one now. There is a highway, beautiful plants are standing here. It gets attracted, so naturally people come and collect this plant. And now, how can we improve this? Now, one of the reasons is, we worked a lot on this particular species. Now, one of the problem for this is, it is a Scrofulariaceae plant, very beautiful, highly advanced group, where cross-pollination is very much important here. Cross-pollination, it is. But since the population is so narrow, although the species is meant for cross-pollination, even from this plant to this plant, if there is pollination, if pollination goes on, it is as good as self-pollination, because the population strength is so low, hardly some 20 plants are there. So therefore, this has induced what is called genetic drift in the species. Until and unless we increase the genetic diversity in this plant, we will not be able to save this species. Now, genetic diversity, how can you increase? You have to bring any other population from this so that you can hybridize this so that the genetic diversity. But unfortunately, so far, only one more population was known in Pakistan, the other Pakistan, and then that was difficult for us to bring. Now, recently, the Jammu area, they have discovered one more population of this. Perhaps people who are all interested, they should try to plant this also and allow this hybridize so that the genetic diversity increases. That is one way of collecting. I think these are all the plants that habitat is being completely lost. Now, ephedra, for example, in the Himalaya, it is one of the surrogates of Soma also. Please see this. It is uh, Somalata. It is also called the local names. They call trade name, call it is Somalata. The entire plant is traded here. In this case, they collect the entire plant and sell it. So therefore, this has become, the threat is harvest for medicine, over exploitation and traded. This is the one. This is how it looks in the natural habitat. It is one of the species of the cold desert areas. Cold desert areas, there are two kinds of rarity in plants. One is anthropogenically genetically rare, and another one is naturally rare. Anthropogenic rare plants means because of the human threats, human-induced threats, the plant becomes rare. Whereas naturally rare, when you say naturally rare, means these species are existing for thousands of years, thousands and thousands of years in a limited area, because Trans-Himalaya, I told yesterday, I showed that area. Trans-Himalaya is extreme north where there is high, harsh climate and all that. So it is in this area, only this, the Ephedra grenadiana comes. So therefore, it is a naturally rare species. There is no threat for naturally rare species. But today what has happened is collection of plants. If it is left alone, absolutely nothing happens to this species. But because of over collection of this particular plant, this plant is becoming rare. Now this is Kutki roots. I told this is the Picrorhiza kurua. This is again a critically endangered species, Picrorhiza kurua, one of the very important plant. And in this one portion, only 1 centimeter or 1 inch you can cut. I'm showing by arrow. 1 centimeter you can cut and put it in a bucket of water. Put it in a bucket of water, and 1 spoon of that water you cannot drink because it is so bitter. So bitter you cannot drink. It is very bitter. And these plants are said to be very effective remedy, apart from fever and many other things, diabetes. Now, we should think of such plants. I can even say this could be equated to the modern Sanjeevani. This is one of the modern Sanjeevani plants. What is the problem for threat here? Harvest for medicine, loss of habitat, over-exploitation. These are all the ones. Of course, the local name I have given. Anyway, don't worry about the local name because it varies from place to place. Particularly here, traded parts are roots. When you harvest the roots, the whole plant is automatically going to die. So this is one of the threat. And unless you cultivate this plant and multiply it in your own area, the plant is going to be completely endangered. This is, of course, from Chitrakoot. This is a parasitic plant, but having highly medicinal plants, scrofulariaceae plant. And monoculture in many places, in Andaman, for example, monoculture is going on. Original forests are removed and they have planted this oil palm. You can see this oil palm, how densely they have planted, and they are harvesting for the fruits, for oil, for its oil. Now, whatever it is economically important, but it is planted in such areas where there is dense vegetation, now that is being. So monoculture in Andaman and Nicobar Island is going to become a greatest threat to the habitat that is going on. So therefore, today I feel there is no option for us, no option left for us except protection of these threatened habitats. Anthropogenic factors, therefore, have rendered many plant and animal species threatened which are, there could be rare, vulnerable, endangered, critically endangered. Therefore, conservation and adequate protection of these threatened species from extinction must be on high agenda on biological researchers in all countries, so that sustainability of these species is ensured. There is no option now except for this, because there is no time left with us. Now, hence conservation is very essential. Conservation helps as a matter of insurance necessary to sustain and improve agricultural forestry. as an investment to keep open future options, as a matter of moral principle, even whether you require a plant or not, you have no right to kill a species. That is the one, as a matter of moral principle. Hey, what will you do with this? You kill it? No. Every plant is important. Every species has a right on its own to live on this world. So therefore, as a matter of moral principle, we should try to allow the other species also to survive in this world. The indigenous system of herbal medicine play important role in the life. So that is another thing. Many of the ethnic leads are attached to the resources, and several medicinal plants found in mountain ecosystem has vanished due to uncontrolled collections, and their habitats need to be protected. Now, there you see the world over, many international agencies today, they are all very much at conservation. It is not only in India, the whole world is looking at conserving this. And following the Stockholm conference, the IUCN formulated World Conservation Strategy. I think that's the one in 2002, particularly the CBD, after the CBD was established, they adopted what is called Global Strategy Plant Conservation, GSPC, we call commonly, it's called GSPC, updated in 2010 with the aim of halting the loss of plant species world by 2020. This was the one at that time. They fixed 16 targets. See, in the so-called Stockholm conference, they fixed 16 targets, which we call Aichi targets. They are all called Aichi targets. And the target time was fixed was 2020. It is 2021 now. Even one target we have not been able to achieve. Remember now to where we are there now. 16 targets are there, and to be completed by 2020, even one target we have not fully achieved. So this is where we stand. And the GSPC has three main aims. One is conserving plant diversity, both in situ and ex situ. Another one is using plant diversity sustainably. So it allows using the plants sustainably. What is a sustainable utilization means? You use it the way you want to the present generation, but at the same time make sure that the species is survived for future generation also. So you should, that is what is called everything you cannot harvest immediately and fill your belly. I think you should allow to the future generation also. That is sustainability. And building capacity for conservation of plant diversity. Now this is lacking in our country. Now we need to develop capacity building through training program and conferences like this, what we are organizing. I think there's a need to this. These are all the IT targets. Forget it. I think we have not been able to finish even. This was in Tokyo, IT targets. None of this we have been able to achieve. Now, IUCN, again, world's most comprehensive agency, it has given an inventory of global conservation status on plant and animal species. And IUCN also has produced or given a set of quantitative criteria to evaluate the extinction risk of thousands of species. And it is the most authoritative guide today. Whatever the IUCN has given us, it is said to be the most authoritative guide for us to assess our biological diversity. So you can always use that as a yardstick and assess your biological diversity. And the Red List Committee oversees the guides and works of, I think that India, we have also this, I think it is going on. These are roughly the IUCN categories. Please understand, extinct in the world, that means we can't do anything now. It is in the completely wild, it is extinct. You can't do anything to save this. And regionally extinct, there may be some species only regionally extinct. Say, for example, a particular species is present in the northeastern region, Gynocardia assamica. It present only in Assam. Now, the Assam region, it is completely extinct. The plant may not be available now. Somewhere this plant has been planted in some other place. It may be growing somewhere. Natural habitat has gone, but that kind of categories are regionally extinct. Now, we can't do any of these things, but these three are these colored ones which I have put here are very important for us. Number one is critically endangered. What is critically endangered? What is endangered? What is vulnerable? What is near threatened? These are all the ones only we would like to. What is critically endangered? A taxon is critically endangered when it is facing an extremely high risk of extinction in the wild in the immediate future. I told the example of the Erimostachys superba near the highway. It is severe risk is there. So that kind of species are critically endangered species because anytime the species may disappear. And now endangered, critically endangered. Endangered means it is taxon is endangered when it is not critically endangered, but is facing very high risk of extinction. These are all the definition given by IUCN for us. So you can apply this to your own local flora. Suppose you are writing a flora of any of your region, you should apply to assess the status of every species that you are working. Now, without assessing the status of all these species, if you write a flora now, it is said to be incomplete. We have to give it is completely. Now, vulnerable means again it is another category when it is not critically endangered or endangered, but it is facing high risk of extinction in the wild in the medium or near future. I think it is likely to. Say, for example, the species is going and there is a proposal that we are going to construct a huge dam and that the area is going to be completely immersed. So that possibilities are there, or we will always say these species are vulnerable, it may disappear. So that's why we need to protect such kind of plants. Now, there are also data deficient means we do not know, we have absolutely do not know. Many of the species are known by type locality. What is a type locality? When a species is described some 100 years ago or 200 years ago, based on a particular herbarium specimen, he makes, I don't know, I will not be able to talk nomenclature, but based on a particular herbarium specimen, he gives the description, and these species are known only by the type locality and not beyond that. No other person has collected. And we do not have accurate data, sufficient data. These are all data deficient species. Not evaluated means we absolutely do not know any of those things. Now, IUCN has evaluated globally, it is globally assessed several species. Now, 81 is extinct. And it's a big 81 species is today considered extinct, and 63 as extinct in the wild. What does it mean? Extinct in the wild, but one or two plants are botanical. In some garden, it is growing. Somewhere in a remote botanical garden, they are growing. So these are all 63 as extinct in the wild. What we do, we can't do anything for this. But for this, at least immediately, we should try to collect seed or multiply all your biotechnology technicals. You can use it and see that species are multiplied, and if possible, try to put it back in the near natural localities and monitor it over a period of time. See that the species survives. So this is one we can do. I think we can put our efforts. 3,947 as critically endangered worldwide, 5,700 as endangered. I think this is global. Let us not worry much about this, but understand that there are a large number of species which are all critically endangered. What is that Indian responses? What is that we have done in India? Now, number one is India, fortunately, is a signatory to all international conventions. So all convention, whether it is a Ramsar Convention, it is a CITES Convention, whether it is a CBD, all convention, India is a member. That means India has great concern towards conservation of its biological resources. So at the government level, we are really seriously concerned to protect our biological diversity. So that is signatory. We are all signatory to all conventions, that you understand. Biological diversity bill and national and state biodiversity boards for all. Today in our country, following the CBD, we have already passed biological diversity bill. I think the details of which I will not discuss because that itself is a one lecture. And national and state biodiversity boards of all states are now without the permission of the national and state biodiversity boards, you cannot go and collect any plants or go for patenting or anything you want to do. You cannot do this. Particularly national, any foreign national, any collaboration with other countries, national biodiversity board is very important. Without the approval, we cannot do. And within the state, you want to do something, state biodiversity boards are there. You should be always in touch with them, take their permission, and then only you can do anything. And then we have included a large number of species under CITES Convention. What is this CITES Convention? CITES is, I think, Convention on International Trade in Endangered Species of Flora and Fauna. I think this, you understand what is the full name. Convention on International Trade in Endangered Species of Flora and Fauna. Under this, if many countries are members to this, I think 178 countries or so are members of this convention now. And under this convention, suppose you every country has listed a set of plants and animals not to be traded. It is a trade. It is mainly meant for trade. You cannot trade some of the species which are listed, and it is binding on all other countries. Suppose somebody smuggles a plant from our country, suppose somebody smuggles an animal from our country, say for example, the elephant tusk, for example, is smuggled, and if it goes to some other country, if that some other country also happens to be a member of CITES convention, they will confiscate that and take that and send it back to the country of origin. It comes back to the country. That means it comes back to India. So there is an understanding between all the countries that each country supports the other country in checking the trade of some of the shortlisted or species which are considered to be a rare species. That I think that is the one CITES convention. I think this you should remember what is CITES, and many places I think this reference comes. And under Man and Biosphere Reserve program, we have declared 18 biosphere reserves. Biosphere reserves are huge chunks of forests. I think that I will just to show some of those things. We have already described described, and 18 biosphere reserves declared, and also the details of management or working plan for all these 18 biosphere reserves also we have been able to bring out and prepare documents for all biosphere reserves. And under national parks, this one, 109 national parks we have been declared, and also we have 340, somewhere around 570 wildlife sanctuaries are also declared. Now, what is the difference between a national park and wildlife sanctuary? National parks are meant for protecting the whole animal, all kinds of animals. Whereas in the wildlife sanctuaries, they are all species oriented. Species oriented means, for example, tiger sanctuary, rhododendron sanctuary, Nepenthes sanctuary, that particular species. To protect the tiger sanctuary, the tiger habitat has to be protected, and by way of this, we protect other species also. So these are all wildlife sanctuaries are there. 27 tiger reserves are there. 26 Ramsar sites. What is a Ramsar site? It is the wetland system, huge wetland systems of international importance. It is not small pond that is growing nearby your area. No, they are all large enough ponds which are having international importance, and that is Ramsar sites. Why it is called Ramsar? Because it is also called the Ramsar Convention. It was in Iran, somewhere in Iran. The meeting was held at Ramsar. Ramsar is in Iran. The meeting was held, and from that time onward, it is called the Ramsar Convention, and wherein it pertains only to the aquatic systems of international importance. And we have declared 17 wetland areas in our country, 15 mangrove areas, 6 World Heritage Sites. These are all the ones great contribution that our country has made, or steps we have taken to protect our the flora and fauna. But still there are many problems that I will tell you. We have also established several gene banks. What is a gene bank? Gene bank is protection of particular species. Citrus gene bank means all the citrus species of particular area are brought and grown or put protected in one place. Citrus gene sanctuary in Meghalaya. In Meghalaya, for example, wild relatives, wild species of citrus are there. So what they have done is that area has been declared as a citrus gene bank, in which all other species and different closely allied species are also introduced. And today that is a citrus gene bank. And we have also done through the Botanical Survey of India, we have also brought out publications of red data books. Now, what is red data book? Critically endangered species of our country. If you want to see that, we give that in the form of a red data book. So Botanical Survey, India is fortunate. We have been able to bring out several red data books we have already brought. That's again credit. Many of the countries are still struggling. They have not been able to bring out red data books, but we have brought already so many red data books, and that shows many of the species are critically endangered. Suppose a species now, you can multiply it and remove the threat operating on that particular species, then that species can be removed from this red data book and shifted back to the green book, so that the plant is safe. That is the one. I think what we need in the coming days is shifting some of these species of red data books to the green book. And please understand, funding research program through DBT, DST, these are all, huge amount. When I was in the committee, also large number of projects we have been approving for whether it is DST, Ministry of Environment, or DBT, for conservation of rare and endangered species, and also habitat recovery program. The rare habitat, if there are any such habitat recoveries are there, that kind of programs, the ministry is very liberally approving. So therefore, some of the teachers are really interested in putting research programs. You should consult some of this in your own area, identify which are the critically endangered habitats, which are the critically endangered species, put up a proposal, get some money, try to do some work. I think that is also, that is a thing that we have done. Also, financial support for establishing a botanic garden, excessive conservation by, see, another program that the government of India has done through the Ministry of Environment and Forest is that we support, again, a substantial chunk of money for the upliftment of botanic garden, not establishment. I think there is a mistake here. It is not establishment of botanic garden. You should have a botanical garden, but uplifting the botanic garden, anything you want to do further, government gives money. But only condition for us is whatever establishment you want to do or upliftment you want to do, you can do, you are free to do. But the government expects in your own region, whatever the critical endangered species are there, those should be introduced into the botanical garden. So our selfish interest is only this: you take the money, but critically endangered species should come in the botanical garden. So when ultimately a chain of botanical gardens are established, each garden has a 10 endangered species. Ultimately, for the country, it is going to make a big difference. So therefore, financial support for establishment, not establishment, for upgradation or making any further improvement in the botanic garden, not establishment. Please, if you want to establish a garden, the government will not give money. But only thing is, you want to do anything further. So putting a fence, I want to do, or putting irrigation facility, I want to do. Anything you want to do, you can do that. I think if you are all belonging to some universities and you are staying in your area has some minimum of, say, 2 hectare, minimum of 2 hectare space is there, and you should always go for some funds so that local species can be preserved and conserved there. Some important conventions, at least you should know. I told India is a signatory for all conventions. What are those conventions? Convention on Wetlands of International Importance, Ramsar. Convention on Protection of the World Cultural and Natural Heritage, Paris, 1972. The Convention on International Trade, CITES Convention, which you call Washington. Convention on Migratory Birds, Bonn, 1979. And the latest one is, this is the one. It has become almost an international law with regard to the utilization, conservation, anything you want to do is Convention on Biological Diversity. Almost all the countries have become members of this country today. Yesterday I told it is the USA is not a member of this convention, CBD. It is not a member of CBD. UK, USA, and another country perhaps is Iraq. These are the 2 countries they are not members of. So therefore, when they go anywhere, they can bring any genetic material and do anything. So we should be extremely careful with such countries, because they, even if they develop something later on, they will not either pass on the technology nor the royalty to our country. I think that is the one. Now, we have done tremendous work in our country, particularly through the Botanical Survey of India under the Ministry of Environment and Forests. We have completed red data sheets. As I told, red data books, 1,182 species we have completed. That's again a good achievement that we have made. 3 volumes, now I think another 2 volumes have come. 3 volumes of red data books of Indian plants have already come, which cover 609 species. And we have completed, compiled complete list of Indian plants in IUCN Red List. So as per the IUCN Red List, we have made a complete list of Indian plants that is in the form of a book already published from BSI. Now, in the red data book, estimated number of threatened species is 1,500. And so far, we have already covered 1,182 species, and red data sheets in the form of red data book, we have published 708 species we have already completed. How to assess or identify rare species? Now, this, if you are working in a small university or a college, now in your own area, how to assess or identify rare species. Now, I must know which are the rare species in my own campus. That is either scrutiny of herbarium specimens in large national, international herbaria. Yesterday, I made a reference about this and I told how you can assess the rarity of a particular species making use of the large herbaria. You go to the herbarium, yesterday I gave the example, if there are 100 people who have collected, only one person has collected only one particular species, only one sheet is there in the herbarium, whereas all other species, some 200, 300 sheets are there, only one is there. That indicates it must be extremely rare. I think this is the one. And following this, you go to the field, you confirm it. That is the one. Scrutiny of literature, of course, flora's revisions. And actual, this is most important actually, actual field surveys. It's just qualitative or quantitative, whatever you want to do. Actually, suppose you go stand there, everywhere you see Parthenium, everywhere you see a particular species. In between, only one species somewhere, say, an euphorbia plant, or somewhere the Solanum indicum, some plant is growing here and there. So in your entire campus, all other plants are there, only one plant is there. So one or two plants are there. You can qualitative, it's immediately qualitative approach I'm telling. You can make guess that this is very rare plant in this region. That is particular for your region, that particular. So that is how we assess the rarity. It could be for a small area, it could be for a large district, or even for a state. The assessment has to be done later on. Now, also understand, yes, plant could be extremely rare in your area, but when you go and work in the state, the plant may be abundant elsewhere. So this, you see, there is again a yardstick for assessing the rarity of a species. For a smaller area, yes, the plant which you have listed is a rare plant. But for a large area, but if the plant is very rare, that should be taken. Now, we normally take one unit as the botanical province. One botanical province we take in the entire botanical province, how many times it is represented, how rare it is, and all that. That is by the qualitative. Now, quantitative, if you want to do, yesterday I told you should throw the quadrats, random quadrats here and there, and assess that. And there are also other definitions, criteria for determining native plants with 3 or fewer locations is rare. Native plants, that means rare in our own plant, endemic plants also. Native plants with 3 or fewer locations if in a given region. So in your entire state or in your district, the only 3 or fewer locations are there. Yes, it is a rare plant. That is by a definition given by Reed. Known only from one population, Siddhal et al. See, they say only known only from one population. I gave the photograph of Erimostachys superba. It is known only one population. It is extremely rare. So therefore, we can say that is another criteria to say that this plant is very rare. But you should make sure the other populations are not available anywhere else. You cannot immediately, you cannot jump to the conclusion, I have seen only one. You may not have seen, but others might have seen. Therefore, you should make sure in while making or deciding the status of a particular cases. And also circumstances which causes changes in the frequency and distribution of species. Now, frequency and distribution of species, it goes on changing. It is not a static thing, but it is dynamic. It is demography always of the species goes on changing. A species, I will say, is it has no threat. If I have to say no threat, my criteria to say is that over a period of 5 years, you should observe if in the population, if there are say 500 plants in the population, that should not change. Either it should not come down or it may even increase. Then it's a safe species. But first year 500 species comes to 450, next year 400. That means decline has started. So we should always observe this, whether it is a safe species. That means the number of individuals, the depth of individuals, and also the production of new individuals should be more or less same. Our production could be more. So these are all considered as rare, safe species. So methods, of course, there are 2 types of conservation. One is in situ conservation, ex situ. I think this is very simple now. You all know this. One is in the natural area where it is conserved. Ex situ is take away from the natural site and put it in botanical garden or some other place so that you can protect from your side. In situ, I have told in India, protected area network, all man's biosphere reserves, protect the wildlife sanctuaries, national parks, and gene sanctuaries. All these are all in situ protected areas. Ex situ, there are two types. One is a traditional way, the sacred forest, for example, is one. And the other one is botanical gardens, seed banks, field gene banks. And modern method is the cryopreservation or DNA preservation, gene preservation. These are all the ones modern. These are all the methods of conservation that we have. Right. Now, of the 10 categories of protected areas suggested by IUCN has protected 10 categories. India, we have declared 9 types. We are already here. Wildlife sanctuaries, biosphere reserve, world heritage site, reserve protected areas, village panchayat forest, private, all these things are there. So that means India has shown keen interest in conservation of protected areas. And this slide, I do not know those of you are interested, how much area we have covered. So in our country, how much area we have covered? In national park, there are 103 in our country, and area covered is 40,500 square kilometer. I think this is the one. That means a huge chunk we have covered. Similarly, wildlife sanctuaries, there are 543, conservation reserves, protected areas, 749, tiger reserves, 50 tiger reserves. This is a good amount that we have already protected, huge amount of forest lands, forest areas. By way of these areas we have protected, and that is credit to our country. Yes. Now, biosphere reserves, see, in the world over, there are 669 biosphere reserves spreading under 120 countries. In India, we have 18 biosphere reserves, of which 9 are in UNESCO chain. That means international importance, UNESCO chain. These are the Nilgiri, Gulf of Mannar, Sundarban, Nanda Devi. These are all the ones that we have done. So Indian contribution towards conservation of biodiversity. These are the ones, biosphere reserves, scattered in different parts of the country. You can see, I don't want to read all this. So these are all the areas where we have scattered different biosphere reserves we have declared. National parks, at present, there are 109 national parks, and which covers roughly 37,534 square kilometers. That means 1.14% of the country's geographical area. Madhya Pradesh has maximum number of national parks. 9 are there. Sikkim with 25% area covered under national park represents the state with minimum and maximum area covered under national parks. I think these are the ones you can see here. In Madhya Pradesh, for example, large number of national parks are there here. That means it is more scattered here. So that may see what I want to say is that these are all the areas where we have protected lot of species which are supposed to be critically endangered. We have already covered in those. Now, what needs to be done, I will come later on. Now, wildlife sanctuaries, I told 543. Andaman Nicobar has the maximum number, 96 wildlife sanctuaries are there. They're all species-oriented conservation sites. Now, World Heritage Sites, there are 6 World Heritage Sites in our country: Manas, Kaziranga, Koildev, Sundarbans, Western Ghats, and Great Himalayan National Park. Now, these are all natural heritage sites. That means you cannot make World Heritage Site, means it is an international import again. For conservation purpose, you get funding from the international agencies. That is another important thing. But only thing is, you cannot develop the area the way you want unless there is a committee, the heritage committee or the natural heritage committee. They will oversee the development. You can't do anything. If you are a convention, you are a member of that convention, you have to obey to that. So in the Western Ghat is also coming under that heritage sites committee. India has 26 wetlands. I told India has 26 wetlands declared under Ramsar sites. 26 areas are there. These are some of the important ones I have just listed in different states in West Bengal, East Calcutta wetland, Dipor Bhil in Assam. All these are all having great importance. Renuka Lake in Himachal Pradesh and Chandratal in Himachal Pradesh. These are all international importance. International importance means it is not small ones, and the anything development or you cannot change on your own. India cannot take any decision. No, I will remove this and make a big building here. No, you cannot do any alteration and all that. They are all of international importance. I think there is a committee to oversee all this. Now, gene sanctuaries, I told native diversity of specific species. I told citrus and musa. I told an Nepenthes sanctuary. I told rhododendron sanctuary in different. These are all species what India has done. Now, keeping this, what will now come to threatened habitats. Now, what is a threatened habitat? Threatened habitats are those areas which support unique species or ecosystem, but have come on road to destruction. Now, there is the whole habitat itself is under threat. Say, for example, wetland systems. Many of the wetland systems have become, because water is drained for agriculture purpose. There may not be any water. The flora and fauna are dying in that system. So these are all the wetland systems. Arid zone, cold desert areas. Yesterday I told this slide, cold desert areas. They are all highly fragile ecosystems. Highly fragile ecosystem, although they are naturally rare flora. There may not be any threat. If you do not go and alter anything, even for thousands of years, there won't be any problem. But since now human disturbance is so great, even the cold desert areas need to be protected very carefully. I think this is an high altitude alpine areas. High altitude alpine areas are equally fragile in the Himalayan system, and mangroves, freshwater swamps. Myristica swamp is a good example. Unique habitats of Nepenthes, Siroi lily in Manipur. These are all best examples of threatened habitats that we have got. Now, which are all those threatened habits? I told sacred, for example, sacred forests, they are all the death traps now. It has become, I have told, wetlands and swamps, Matharanwala, Gola Tappar in Doon Valley, Myristica swamp in Western Ghats, mangrove swamps almost in the southern India, 6,700 kilometers. Yes. Now, Matharanwala swamp in Doon Valley, I don't want to go for definition, but understand it is it represents one of the serial stages from open water to closed forest canopy and lead to the development of tropical rainforest. I think that is still in the stage of developing a rainforest, moist rainforest, just like we have in the southern India. And it formed a continuation from west to east, but now only Kaziranga and Doon Valley, only the two packets are left. And large number of species, for example, it includes trees 34 plus, shrubs 25 plus, climbers 193. There is a dense vegetation. If this habitat is lost, all these species are going to disappear. So therefore, this is a typical picture of the swamp vegetation that we have in Doon Valley, Matharanwala swamp forest. And if water is drained permanently, now what is going is the water level has already come down. Only during monsoon, some amount of water will be there. Otherwise, the agriculture, the surrounding areas, the agricultural development has taken so much that the water is being drained. With the result, the swamp forests are also disappearing. Threats to this valley, draining of water, I told. And many of the species here like Centella asiatica, Bacopa maniri, Roripa, Nasturtium aquaticum, Diplasium esculum, all these species typical of the swamps are disappearing from that area. Added to that, today, invasive species like Eupatorium, Lantana, Parthenium have also invaded the swamp areas, and that is, they are threatening this. Myristica swamp, I told, this is Myristica swamp that we have in Uttara Kannada. It's a wonderful, it looks like a mangrove. See, you can see here the stilt roots. Stilt roots is the characteristic of mangroves. Yesterday I told and showed some photographs also. Now they are so hard. The same thing here. In this case, there are large number of stilt roots coming up, anchoring that. And typical, always permanent water is always there. These are all typical there. And they are all Myristica species. Since they are all, majority of the species are Myristica, they are all called Myristica swamps. So they are characterized by stilt roots and knee roots. These are all the ones, the knee roots, stilt roots. These are all the ones which comes. And there are 63 species and of 130 flowering plants identified, which are all endemic to Western Ghats. 130 flowering plants identified from these swamps, which is said to be endemic to this region, right? Now, which are the dominant species? Myristica fatua, Gymnocranthella coronarica. These are Conerica. These are all the ones which are all the dominant species restricted exclusively to Myristica swamps. We don't find this anywhere else. And 4 genera comprising 15 species of Myristicaceae are found in India, of which 3 genera, 5 species are endemic to Western Ghats only. Myristica swamps reveal high diversity of flora and fauna. KFRI, particularly Kerala, they have listed 65 tree species, 72 species of shrubs and herbs, and high levels of species diversity, abundance of many animals like reptiles, amphibians, which are all recorded, which these are all Now the whole, it is not the species are threatened, the habitat itself is threatened. And since the habitat is threatened, there is a threat to all these species in the coming decades. Myristica swamp, yesterday I told, it is a living fossil community. Now, what is a living fossil community? Once it is a very primitive origin. Once it existed, the entire area, the age is so archaic that it has been spreading everywhere. But today, it is confined to small patches here. So that's why I put it in inward, a living fossil community. Myristica swamp is a living community and is regarded, Myristica is regarded as one of the oldest tree species. It is one of the oldest tree species. The oldest, not individuals I am telling, the age of the species I am talking is one of the oldest. Face severe anthropogenic threats and Myristica species are released plywood industries earlier, while today swamps are drained and converted into areca palm and teak plantations. So this is a major, of course, they want economy, people want to develop economy, we want areca palms also, but you see the malady is some of the unique swamps which have been surviving for millions of years are today threatened with extinction. Now, some of the other swamps are this. Now, Sundarban, Bhattarkarnika, Godavari, these are all mangrove swamps. That is the Myristica is a freshwater swamp. These are all the mangrove, the saline swamps. These are all the saline swamps. Pichavaram, Baratang, Island mangrove. These are all the ones. And Shola forests equally. Shola forests also is a, I will call a threatened community in our country because a lot of disturbances are going on in the Western Ghats. Already hottest hotspot it has become. I call this also alternatively called tropical mountain cloud forest or simply cloud forests. I call this also as a living fossil community. The sacred forest, the Shola forests are also like this. Now, members of Laraceae, Rubiaceae, Symplocaceae, Myrtaceae, Myrcinaceae, Olacaceae are predominant in these forests, in the Shola forests. Very typical rubus, Daphne-phyllum, urea, rhododendron, berberis, or some of these are Himalayan elements. Rubus, Daphne-phyllum, urea, subtropical rhododendron, mahonia, berberis are the Himalayan elements. Now, understand in the Western Ghats, there are a large number of Himalayan elements. Now, you may be wondering how these Himalayan elements have come to the Western Ghats. Now, there are 2-3 definitions or the phytogeographic discussions are there. One definition says that during the Pleistocene glaciation, there was heavy glaciation during the Pleistocene era in the geologic past. The Pleistocene glaciation, it is a global phenomena. That Pleistocene glaciation was a global phenomena. And during that time, the temperatures lowered so much and it existed for thousands of years. During that species could not tolerate the extreme cold. So gradually, many of the species started moving towards the equator. Equator means it is South India. They caused this, so gradually moved. Moved means it is not simply walking like any animal. So it moves several thousands of years it has taken. They gradually moved towards the equator. And with the retreat of this Pleistocene glaciation period, after the retreating of this glaciation period, what happened again? These species could not go back. Rather, they climbed the mountain tops. So therefore, in the Western Ghats, many of the species in the mountain tops are similar to the species that we find in the Himalayas. So each one of these species is called a Pleistocene relic. Rhododendron is a Pleistocene relic. Mahonia is a Pleistocene relic. Berberis is a Pleistocene relic. If somebody says, what is a Pleistocene relic? You should remember this. There are a large number of Pleistocene relics, not only in the Western Ghats, even in Adams Peak in Sri Lanka. Adams Peak, Sri Lanka, you go, large number of plants are there. It is almost like our own Western Ghats. So that all these are all Pleistocene relics. We should have always remember this. Now, Janaki Areal Patra, it is a unique community. It grows, in the rocky beds, but steep, areas like this, it grows. It is highly important medicinal plant, and people are collecting plants from this. I think we should try to protect this. Now, conservation of threatened habits should be a collective responsibility. It's nobody's, it is not mine, it is government of India will do, or forest officers will do. It is a collective responsibility. Everybody should do this because only one earth, we have only one earth, that is one. the World Heritage Trust, Biosphere Reserve Network, Convention and Plan, all are struggling to do this. It is a unique, and the local people also should come forward in helping the conservation of the biological diversity. Now, this is one grass, Habardia heptanura. This is one grass which was presumed to be extinct in our country. It was presumed to be extinct. It's a rare grass. Now, you may ask this question, what is the use of this grass? There is a plant is growing which has no economic significance. As I told, it's a moral ethic. Every species has the right to live. We cannot kill. Now, this was presumed to be extinct in our country. Like in our Dr. Yadav, he has been from Kolhapur University. He has been doing very good work in the conservation. He has been able to identify this plant in not only in the Jog Falls, that is the type locality, not only in the Jog Falls, he has been able to collect this plant. And today he has rehabilitated in many other localities. Today the species is saved. Now from extinct, we can bring, you can say that the species is surviving in other localities. Even you can shift it to Green Book. I think that kind of work that this fellow has done, it is really creditable. Now, look at the habitat. It grows in such habitat where water is dripping, very unique habitat, and he has been able to introduce plants in all these localities. In such localities, he has been able to bring and introduce. Today, Hapardia leptophyllon is growing all these areas. Amba Ghat, Agumbe Ghat, Ambuli Ghat, all these areas, very profusely the plant is growing. That means we have been able to save one species from extinction, which was presumed to be extinct. Now we have been able to bring. This is real conservation. We have to monitor this. And suppose I introduce 20 plants here. Oh, I have introduced 20 plants and they are safe. No, you have to observe this. Next year, the 20 plants should not become 18, third year 15, 10, like that. The decline in individual number of individuals should not be there. If the same population continues, if I introduce 20, every 3-4 years, it should be 20. That means the population has stabilized. So that thing you should make sure that is real conservation. I think that you should understand, which requires, I think, a lot of simply you cannot do this. If you want to conserve, you have to, you cannot bring all the plants from wild. You should multiply in your nursery. Suppose a rare plant is there, you multiply that in your nursery, thousands and thousands of plants, and then shift it to the area where you want. Look at this. This is again the very important medicinal plant belonging to Liliaceae. All these things are Camptorrhiza. All these are very important plants. And we see the whole group there. I think I will appreciate this group. They are not sitting and enjoying here. In the type locality, in the, look at this, the hillocks of the, I told about Khas Plateau yesterday, lateritic soil. In such kind of lateritic soil, this species grows. The whole persons, they are all the broad, they are specific species they are brought, and gradually they are introducing, go on, keep putting. So, but by the time, I think they will be able to complete after after another 5 years, 10 years, all these species will establish. Now, in the cold desert, similarly, I think there are large number of critically endangered species that we need to protect only in the cold desert areas. You cannot bring these plants and multiply in some other habitat. This is the problem. We cannot bring because they are all adapted to that particular climate. We have to multiply in a nursery established there only and try to multiply some of these plants there only. Nepenthes sanctuary, now we have declared it as a Nepenthes sanctuary, and this is a prohibited one. Nobody can go and collect, but illegally, most of these plants are being collected by university teachers who go with a big batch of students. The moment they see this plant, every student will collect immediately 2, 10, 20 pictures of this. The whole area is destroyed. I think this is what is going on in the Khasi Hills of Meghalaya, Jaintia Hills of Meghalaya. All these plants have become critically endangered. In fact, when I was there, I took several photographs and sent it to the forest department. Look, it is a conservation area, and you said it is protected. Any teacher, student, batch comes and they are collecting this. I think this is happening. Now, I do not know the present status, how many plants are left, but this is going to be critically endangered. Now, this is again Yadav's work I have taken from this. He described one plant, a highly important medicinal plant. This is a nala. You can see this. This is Crinum malabaricum, a new species. This is a nala. Look at the long leaves of this. It is a wonderful discovery he has made, and highly important plants. And look at this, our students removing their shirts and all. They have gone into the Nala and collected this plant. That shows real botanizing work is going on. I think that's the one. But what I want to say, this is the unique. This is only occurring in only one place, somewhere in Kerala region, only one in Maharashtra region, only one place, right? They have collected this plant, described it as a new species, and this is the habitat of this, not habitat. Yes, habitat of this is a very threatened habitat. Today, look at this. This is what has happened is this, in the name of development, the entire plant species is completely destroyed. The habitat is completely lost. The species is gone for all the time. Now the species is not available, except some of the plants that he has protected in the botanic garden, they are available. I think that's what is going on. Ex-situ conservation is botanic garden. I told about the botanical garden, and I told under the assistance of botanic garden, the government gives money. Perhaps you can all, some of you should be able to submit if you are all belonging to universities and you want to really do something, submit a program, get some money and try to go for developing botanical gardens in your own colleges and university. And in the botanical survey, of course, is a big chain of botanical gardens we have, and also orchid area we have. And under Botanical Survey of India, large number of botanical, I do not know how many of you have visited. I think we should visit. This is one tree actually. The whole thing, it looks like a forest, is one tree of the ficus. That is the one, and you don't know which is the main trunk here. You can see it is a forest. When you enter, it is completely a forest. And we have a large number of gardens under the botanical, in the Botanical Survey of India. One is the Howrah Botanic Garden, AJC, Acharya Jagdish Chandra Bose, that is the one, Indian Botanic Garden, and then associated botanic gardens at Itanagar, Barapani, Allahabad, Dehradun, Paudi, Kirsu, all these areas, associated botanic gardens of the Botanical Survey of India we have, and they are conserving large number of endangered, critically endangered species. In addition to that, a few orchid area are meant for protecting only the orchid species. So orchids are also there. Under the university system, also there are many other national botanical gardens like Malabar Botanic Garden, University Botanic Garden at Calicut. It's a beautiful garden. Calicut Botanical Garden is a very beautiful garden. University Botanic Garden, Shivaji University, Kolhapur. I think this is a very good one. I think you should all visit this. And the University Botanic Garden, Calicut. These two, I feel extraordinarily, they have done wonderful work. I think you should visit if you happen to visit some of these areas. And in our own Bangalore, we have Mahatma Gandhi Botanical Garden. Now it is considered as a lead garden, which can help other smaller gardens in supplying seeds and other things for developing their garden. I think that we are doing that one. And Desert Botanical Garden, University Botanical Garden, MS University of Baroda, some of these gardens are there. Now, these are all some of the botanical gardens. Again, I just don't want to go into that. And some of these species, the rare species which we have listed here, a huge list of species I don't want to read. These are all critically endangered. They are all luxuriantly growing in the botanical garden today. So that means at least one thing we should feel happy that they are growing. Now what is required is that we should try to multiply these species. And as I showed some of the nurseries, huge amount of seedlings you should raise and take it to the natural or near natural habitats and try to sow these plants so that, and monitor over a period of 5 years at least. We may not be able to do long, at least so monitor over a period of time. And if they are taken care, yes, we are very happy. And each one botanist saves one species in his lifetime. That is the greatest contribution as a botanist we are doing. I think that's, you should keep it in your mind. Threatened species in Botanic Garden again continues. These are all the Erimostachys, Sasuria, Freeria indica, Cypripedium, Trachycarpus tachyle is a very important form. It's a palm. It is an important farm, and we reported it as a new record at sometime back when I was in Nehu. And this is Trachycarpus. The close relative of that occurs somewhere in Indonesia and all that. I think that's another important one. And some of these important orchid area and nurseries we can see here. Now, yesterday I told about this, so I will skip now. I think my time is also getting, yes, it is already 12:30. Sacred groves, yesterday I told, this is another conservation in tradition. Sacred groves, yesterday I told, so I will skip this. I told also that the existence of these sacred forests is a matter of time because of two reasons. Number one is there is no biological corridor, I told. and the flora and fauna die in situ. So therefore, they become death trap today. The other reason I told is the belief attached to the sacred forest is eroding. So therefore, we should now strengthen that belief. Wrong or right, we should strengthen that belief so that the species survive, the habitat survives for longer time. The millions of years, they have existed for millions of years. Now, in our present century, in our present generation alone, there is a threat. All these years, there was no threat, but now there is a threat. I don't know how well, how good you will be able to protect this, because some of the younger generations here in the audience, I think you should take interest and see what you can do, what best can be done to protect the sacred forests in your own area also. Now, some of these species which are all occurring in the sacred forests, again, some of these species, and they are all protected because of some beliefs. Yesterday, I told about some beliefs. Say, for example, eager marmalas is offered to Lord Shiva. Oh, it is a sacred tree, I will not cut. Michaelia champaka, favorite with South Indian women, and Ficus bengalensis connected with Lord Krishna. So therefore, some of these species are protected because of some belief attached to. That is what is called tribal wisdom and conservation of biological diversity. I also told about the 27 nakshatras. If you know your nakshatra, please see which is your plant. If you are Ashwini, Stricnas naxwamika is your plant. You go and worship your Stricnas naxwamika. That means you should not cut that plant and prosperity will come. That is the belief. That is the belief. Now, if you believe in this, at least you do that, worship that and see what happens. But whether something happens or not, the species will be protected. That is exactly all these 27 nakshatras. You can see whatever is your nakshatra, the species I have listed, each one is listed. That is again a tribal wisdom that we have. Then planets, each planet, Calotropis gigantea, for example, is a species attached to sun, Butea monosperma to moon, Acacia to Mars, Ficus religiosa to Jupiter. So all these associated with plants, and they worship that. Now, in your own small area, you can do your own protection in a small area like this. If in your botanical garden you want to establish, for example, this is the area that was given to us when I was in CMAP Bangalore at one time. When I was in CMAP, I took a small program from the Ministry of Environment and Forest. I told this one that strengthening the botanical garden they give. So under this program, I took this program with a small amount, and we collected all the tree species, endemic tree species, not the herbaceous ones, because herbaceous species are also important. But what I did, herbaceous species every year we have to rejuvenate. Somebody has to take care of. So therefore, I selected only arborescent, endemic tree species of the Western Ghats, and more than 100 species we collected. And it's a big forest today. If you visit CMAP, you can go and see. It's a nice forest of endemic species only. Today, we have all of them are flowering, fruiting, everything is going on. So endemic species. And we also named this as the Srishti. We called it the name Srishti we have given. And I don't know whether we have some photographs. No. So what I want to say is that in your own area, you give your own name and invite some of the VIPs from your own area, get inaugurated and get the planting done on Environment Day or some occasion. Try to do some of the planting done. They will be happy. They will be able to help you in funding, in getting funding and all that. But ultimately, what is more important is conservation of some of the rare and endemic species of your own region. So some action for conservation of biodiversity, assessment of threatened species, number one, seek international cooperation, assessment of habitats of threatened species, and assess the major threats to species. That is also we should know. If you have to overcome the threats, we should know what are the major threats for that species. That is the one. Explores the possibility of their in situ conservation. Yes, if it is possible, in situ is preferable. If not possible, take it and go to ex situ conservation. And rehabilitate in natural or near natural habitat and monitor this population. I told that the Ministry of Environment or DBT gives enough funds. At least when I was in the committee, I used to strongly recommend this only on the condition that at least minimum of 500 plants we are going to put it back in nature. That was my condition, that 500 plants you should go and put it back. That was the, not simply you bring 10 plants are there in the wild, you bring 8 plants and put it in your botanic garden, say I have conserved. No, this is no conservation. Conservation is ends only when you multiply the species, not from the wild. You should multiply the species and rehabilitate it in natural condition, and if possible, monitor the population over a period of 1 or 2, 3 years, so that you can do that. So that is the one. So that is conservation program. So rehabilitate in natural and near natural habitat, encourage cultivation of important threatened medicinal plants. I think this next class I will be able to talk. Develop agrotechnology of these species. Tribal wisdom, strengthen the tribal wisdom in conservation. I think we should always do this now. We laugh at that, "Arey, devil will come and kill you. Oh, no, nothing will happen." We laugh. Now let us attach importance to this and say, yes, devil will come and kill you. Please don't touch. So that is revitalizing the tribal wisdom, revitalizing their belief is necessary. Otherwise, the sacred forests, it's a living fossil community, is going to be lost forever in the coming days. So let us contribute our bit to save our biological heritage. I thank all of you. And if there are any questions, we can very gladly ask some questions and clarifications. Yes, even otherwise, those of you who want to be in touch with me, will note my email. I will be available on email, and anytime you want to know something, yes, whatever is possible from my side, yes, I will be able to do that. Now, please note, I am not now in any of these committees, fund giving community committees, but if I were to be in the committees, I would have helped many of you to get projects and all that. But what I can do, yes, I will be able to help you. Thank you so much.
Thank you, sir. Thank you so much. Sir, you have again a lecture by you in afternoon. So please,
yes, what time?
Stop sharing the, at 2:45 your lecture is again. So, sir, can you stop sharing the screen? We will have one more photo now.
Okay, okay.
Dear participants, please, can you all start your video once?
Stop sharing this.
These are the memorable moments for us, all of us. For last 14 days, you are with us.
Yes. They must have been bored now, Dr. Ahmad Alimad Khan. They must have been bored hearing only the.
Aisa mat boliye, sir. Aisa mat boliye.
Please let me, I am also a human being. Please understand. See, there is Academies supports the lecture workshops. You know that. Now, what is the difference between a lecture workshop and a refresher course? Lecture workshops are for 2 days, refresher courses are for 14 days. That is all, no difference now. See, refresher courses, please understand, but the other teachers also I am telling, please note, refresher courses are theme oriented. It is not like this anything, like say, refresher course in taxonomy, refresher course in ethnobotany or molecular biology. Refresher courses are theme oriented, number one. Second, what we talk in the morning session should be followed by practical in the afternoon session. So the continuous sitting and listening to the teachers, some 50% will not go in their head. So to avoid that, practical should be there. On this kind of webinars, there is absolutely no practical. All the 14 days, listening. Somebody will speak on fungi, somebody will give on molecular biology, someone will speak on taxonomy. You see, there is no connection. It should be theme oriented. Please remember, it should be, if any of you are putting a proposal for next year, identify a theme and put the proposal on that. I think that would be good. Thank you.
Sir,
yeah.
the time is not allowing us because of now again there is a lot of increase in positive cases and lot of social.
I am not denying. I am just telling that that's why teachers get bored.
Priority. We are also teaching from online only. As a teachers also, we are also feeling somewhat different kind of era. We are living in a different kind of situations. So where we need to upgrade, we should not stop upgrading our knowledge and making ourselves upgrade.
Why don't you say make it reproductive biology or reproductive ecology? Only those specialists, you should, one area you should strengthen. Like this, something talking algae, someone under immediate next lecture will be on molecular biology, third lecture. See, the teachers get different kind of diversity you want to bring. Anyway, that is a different thing left to you. But I am happy to interact with all of you. I do not know from where they have all come. And yes, very nice to see some of them. Good luck to all of them.
You can have your cameras on, please, all of you.
Camera is on, yes? Yes, okay.
Indira ma'am, Subbaraja sir, Rajesh, Abhimanyu, in a different times we have to live in different ways. So this is
Yes.
also one of the way how to live. We are learning now in a different times. When earlier pandemics were there, there were no options. But now in this pandemic, just because of the improvement in technology, we could gather. See,
Arre
we
bhaiya, you are very familiar with the technology, but I am not familiar with the technology. I have to call my granddaughter to
no,
adjust everything.
no. You are very competent. You see,
My
from last,
granddaughter is helping me.
yeah. Anyhow, somebody
Yeah.
will help. You're also learning from
Yes.
somebody else.
So
Yeah.
that's why. Anyway, please. Okay.
Please, Sai Venkatesh sir, can you make your video on? See, sir, here we have in these participants, one just to side of you, there is a Dr. Sinaya sir, he's from Andhra Pradesh, and down to you, Mujahid sir is there, he's from Maharashtra, and Dr. Suresh is there, he's from our own university, he's working in one of the college, government college, and Dr. Deshmukh madam is there, she's from again Amravati, Maharashtra, and down to there you have Pradeep, Dr. Pradeep Kumar, he's from Odisha, and down next to Pradeep sir is Sunil Kumar sir, he is working with our government college, he's also from Giriraj College, and down to you is again Dr. Sudha madam, she is from Hyderabad, and then next is our Vasudev sir, he's Dr. Vasudev is from our one of the college in Nizamabad, and then we
Okay.
have Rajini madam, she is from Tirupati University. So down to you is our next speaker in the afternoon, Dr. Mahendra is there. Then we have one more person, Harrison. He is from again government college, Giriraj Government College in Nizamabad. He's from biochemistry faculty. Then we have Mohammad Farooq. He's again from Kamareddy and he's a scholar of our university, biotechnology department. Then we have Ruhia madam. She's a teacher, faculty from Hyderabad College. Then we have Dr. Jalandhar. You have seen our Dr. Jalandhar. He's from our department. Then Rajesh is there. He's working in one government college and he's pursuing his PhD. Then we have Dr. Abhimanyu. He's from again Amravati, Maharashtra. Then again we have Dr. Subaraja from Tamil Nadu, Vivekananda College, where you had earlier lecture workshops. And we have Sai Venkatesh sir. He's from again Andhra Pradesh. And Dr. Indira is there. She's pursuing her PhD, it seems. So she's from again Osman University, Hyderabad. Then again, one more madam is there, Aisha Fatima. She's from again Hyderabad, one of the college. And Kalyani madam is there. She's also from one of the college. And some more people are there who are not having their camera here. But still, we have a very great gathering. And thank you so much. 37 people are there, but in the
very, very nice to know the different parts of the country they have all assembled here. But let me tell all of these teachers that Dr. Halim Khan is a very active worker. In fact, I have been in the know of this fellow. I think during the last 4 or 5 years, he has been conducting this kind of lecture workshop in his own college, university. Not only is conducting in his college, but he is making other colleges in his own area also to submit proposals and conduct. He is showing very active. So I want to tell you that you should all become active, submit your proposals to the academy in the coming days. If possible, if COVID problem is solved, then we can all meet and discuss and give regular lectures. But otherwise, you should submit your proposal. I am myself conducted, I think, 2 or 3 lecture workshop in his college in person. I think during the last few years, we have gone and met all his faculty, and he's a very active fellow. Now his activity is further increased now. I think that is what I have been observing. Earlier, he never used to sit in the class. Now he sits all through because the responsibility has come on him. So I think that is the one which makes him to listen to all this. I congratulate Dr. Halim Khan and all of you. God bless you. Good luck to every one of you. Thank you.
Sir, we have some more people down. We have here is one of my postgraduate teacher, Aisha Siddiqui madam is there. And I don't know, Ranjani madam, can you later on in the afternoon speak with Swarna madam and ask her to keep the camera on? Sara madam, can you on your camera, video? Ramesh sir, you also.
Indira, okay.
Both Praveen Kumars also, please. Both Praveen Kumar sirs, please switch on your camera. Rajgopal sir, you also. Dr. Srinivas, you too. Gyaneshwar, and Vani madam, all of you, please. This is it's a precious moment. We are in a very memorable moment now.
Yes, you are
Ranjini
taking a
madam,
photograph
please
of all
call
this.
Swarna madam in afternoon so that she can have the camera on, because I don't know how she is not reachable.
No, what is the problem? Why are they not putting their camera on?
Maybe their devices, I don't know what kind of devices they may be using.
Okay, okay. So yeah, now it is okay. Yeah.
Dr.
Okay.
Srinivas, you too, please. Dr. Srinivas, quickly.
Hmm.
Everybody is ready for lunch.
How many total? How many participants total?
Ramesh, please, Ramesh sir.
Oh, 37. Good. Okay.
Praveen Kumar sir, Andhra Pradesh, Praveen Kumar sir, and our own biotechnology Praveen, please, can you? Vani madam, please, quickly you do, yaar, because everybody is hungry now. Dr. Srinivas, please, quickly. Siddipet, Dr. Srinivas.
Yes, why are they tuck karke? They should all put on those cameras.
I don't know, their devices may be not supporting or what, I don't know.
Okay, yeah.
There are still people without camera on. Okay, anyhow.
I would have also become one of them, but my granddaughter is there. She is helping me. That's how it is. Quickly, I'm able to do.
Tara ma'am, 1 मिनट के लिए सिर्फ एक स्नैप, और नीचे के कुछ लोग रह गए हैं इसीलिए प्लीज सारा मैम जल्दी कीजिए.
Still, who is left? Another 2, 3.
Anyhow, thank you. We'll join back again at 1:30. All of you, please join back again with us. Thank you.
My
We'll
lecture is 2:45, right?
Yes.
Okay. Okay.
Thank you. Thank you, everybody.
Okay. Thank. That is the last lecture for me.
Yes.
Okay. You consider me as the last person, eh?
No, we will have validity again. We will have validity again.
Okay. Okay.
Thank you. Thank you so much. Thank you, everybody. Please join back again. We will have some more photos. You will also have the memory, and we will also have the memory. Please. Thank you, everybody. Thank you. We'll end the meeting now.